Sumana K, JSS Aher, India. Now I request Dr. Sumana K to introduce our session chair.
Thanks, Dr. Pallavi. We have the session chair, Dr. Kandaya Pakiratan. He pursued his BSc and MSc in agriculture and also pursued his PhD in university at Sydney. And his area of expertise is in pest management, genetics and plant breeding, molecular biology and biotechnology, plant pathology, nematology, molecular genetics and plant breeding, plant protection, environmental issues. His special interest is in breeding for resistance, molecular characterization and identification of plant pathogen interaction and disease development, chemical defense of plant against pathogen, identification and characterization of gene expression in resistant plants, environmental pollution control. He has edited many chapters and his contributions can be seen in book chapters, many book chapters, and also his publications are more than 100, can be seen in most of the peer-reviewed national and international journals. And he has many awards for his credit. His CV runs to around 14 sheets, where I have just cropped it up for only 2 minutes talk. And he has served as a resource person for many international as well as national seminars. And he has many awards and scholarships for his credit. To name a few: Merit Presentation Award, Best Poster Award in Rahunu International Science and Technology Conference, Mahapola Scholarship Award, etc. He has membership and professional experience in many of the forums as well as platforms. To name a few: full member of Sri Lanka Academy of Young Scientists, chairperson, Education and Research Subcommittee, General Secretary of Jaffna Science Association, etc. And he is a member of many editorial board and also reviewer for many journal and conference. With this brief introduction, I invite Dr. Kandaya Pakiratan to chair the session.
Thank you very much, Dr. Sumana, for your nice introduction. So very warm welcome to the session 4. In the session 4, there is a keynote speak. After the keynote speak, there will be 4 presentations. So first is the keynote speak. The keynote speak is going to be delivered by Dr. Nabil Suyij, is the Director of Alliance of Collaboration on Climate and Earth System Sciences, City of Cape Town, Western Cape, South Africa. This is my privilege to introduce our keynote speakers today. So Professor Nabil Anthony. He graduated from the University of Cape Town and completed his PhD in Cape Town, University of South Africa. So currently serving as a director of ACC ESS, and he has been contributing the developing and training sessions throughout the South Africa, and also he is guiding government, instructing the government organizations to develop the policies and initiatives. And he has been published a number of papers in these high impact peer-reviewed international journals, and he has been continuously delivering the keynote talks and the other invited speaks in many forums. And he has been awarded a Scientific Merit Award for his contribution to the earth and environment. In such capacity, I would like to introduce today's keynote speaker, Professor Nabil Anthony. Actually, he is here in the online mode, and he sent his speak in a video record program. So the video record program will be run by the IT section, and he will be here throughout the session for question and answering. This is IT section. Please, stop.
Good morning. My name is Dr. Neville Swade. I am the Deputy President of SCOPE, and I am the Director of the Access Program Alliance for Collaboration on Climate and Earth System Science based in South Africa. I thank you all very much for the honor of being able to address you. And I apologize that I'm not there in person. I did intend to be there. I ran into some visa problems. So I'm going to now give you a presentation on an overview of the situation in South Africa with respect to plastic pollution. It is indeed focused on thermal, on terrestrial pollution, but I do have a link to plastic pollution in the oceans, as will become apparent. So I will talk about South Africa's national flower, amongst other things. Just for your information, the national flower is the protea, and I know that as cricket lovers, that word protea will strike fear in your hearts in India, but I do understand the situation. Good. So I will cover these points. I'll frame the problem. I'll give you some statistics. I'll talk about the national flower and myrtles. I'll briefly touch on legislation and regulation and research, and then talk a little bit about the formal and informal recycling economy, a bit about what we are doing about the problems, and finish with some perspectives. So I'll start by framing the problem, and I want to point out that South Africa is a very poor and unequal society. We have, unfortunately, the dubious honor of having the highest Gini coefficient in the world. You can see it on the map here with the deepest blue color. And I'll come back to that in a minute, which means that inequality is extremely high. And I'll tell you why that's relevant in a minute. And on the left, the chart shows in blue the South African data, showing that the highest 20% of income owns around 70% of the country's wealth. The red line gives a emerging economy median. On the right-hand side is also the Gini coefficient here between 0 and 1. We're sitting at about 65 and persistently higher than the emerging market average, persistently so, fairly constant over the last 15 or so years, and having increased, in fact. So inequality is, in fact, increasing in South Africa. And then I want to talk about poverty. And when I talk about poverty, I know that we are very familiar with that in India as well. And here, really grinding poverty in South Africa, with about 49.2% of the population living in poverty. The rest of that is a distribution across the country, with some areas of up to 68% of people living in poverty. Where I live in the Western Cape, we have a figure of about 33.2%. This is fairly old data, but I don't think things have changed very much. So why is this important to frame the problem? It's because there's a relationship between poverty and plastic dependence. And these two images illustrate that point. Plastic goods are cheap and durable, and people use them for cutlery and crockery, for furniture, for storage and transport of water and dry foodstuffs, for storage of cooked food, and of course, very importantly, a source of income, which I'll get back to later. On the bottom, you'll see this image of water in these plastic bottles. Now, this is taken around the corner from my house. In fact, was very much an important element of our very famous Day Zero drought, when Cape Town ran out of water and people were harvesting water from streams and from springs all around Cape Town. So that is the point about poverty and dependence on plastic. Now in South Africa, some statistics for you. We produce about 2,500 tons of plastic waste is generated in South Africa per annum. And of that, 70% is collected, but only 14% is recycled. And about 80,000 tons a year of plastic leaks into the oceans. And this is why it's so important to consider terrestrial plastic pollution along with ocean pollution. And so on the chart on the left, you'll see the sources on the left-hand column, imported plastic with goods that are imported, and import and production of of plastic for production and manufacturing here. And on the right-hand column is just a breakdown of what is the fate of that plastic, of which about 1,000 tons is properly disposed of, but a big proportion is improperly disposed of or uncollected, and as we said, leaks into the ocean. So that's a bit of a background of the quantity. Now, this might not seem a lot in the context of global pollution, but I'll show you that South Africa is disproportionate in this respect. Before I get there, I want to make this message very clear, that poorer people produce less of the waste. And on the top left-hand side and the bottom left-hand side are pretty much the same data. It shows the distribution of per capita income on the x-axis and the per capita waste production on the left-hand, on the y-axis. And you'll see South Africa sits somewhere in the middle. But the important lesson, if you look at the bottom part, the bottom left, you'll see that countries with higher income produce higher quantities per capita of waste. And on the right-hand side, here is a breakdown of how our societies produce waste. And you'll see that this point is that simply the lowest amount of waste is produced by the lower income groups, and that's emphasized in the bar graph below. So the message here is that poor people produce less waste and wealthier people produce more waste. And I want to come back to that towards the end. This is just a quick summary of the fate of plastic in South Africa. Again, on the top right, I said 14% of plastic is recycled, and that's just a breakdown of where it comes from and where it goes to. The top left indicates that about 950,000 tons of plastic is mismanaged, and 48% of that is in fact burnt and produces a significant amount of impact on the air quality. The bottom left shows the total leakage that was assessed in 2020, of which about 6.5 kilotons is micro waste, microplastics. And on the bottom right, I've just put a chart here which gives you the breakdown of the kinds of products that produce this waste. And here you can see that plastic bottles are really the biggest source of the problem. And in fact, bags are the second most important of those, plastic bags. And then there's others like food containers, baby diapers, and other sources. Okay. Now, just to get a global picture and see where South Africa ranks, on the left-hand side, I've got a chart which shows what parts of the world produce the most waste. And about 80% of the waste comes from Asia, of which India, in fact, produces about 13% of global quantities of waste. Now, of course, that's not proportional to the population size. I guess that would be much lower if we did that. And Africa as a whole produces around 8% of the world's waste emitted into the oceans, with South Africa responsible for 0.44% of global waste into the oceans. However, if we look at it per capita, you'll note that South Africa, in fact, beats India, and we are position number 11 in terms of mismanaged plastic waste per capita in South Africa, tons per year. So South Africa really does, if you like, produce more waste than is in fact reasonable given our population size. And we are the biggest emitter in plastic waste emitter in Africa. So now I'll talk a little bit about the national flower, so-called. Of course, this is not our only plastic pollution problem, but it is certainly the most visible of them. Very prominent in South Africa. Our actual national flower is the Protea, as you will see down below. Up until regulation began, we were producing around 8 billion lightweight single-use plastic carrier bags per year. And over that time that monitoring took place, plastic bags had been progressively thinning, meaning they were getting lighter and lighter from around 30 microns in 1982 on average to 14 microns in 1999. And then the government stepped in with two measures to try to manage this problem. One was to regulate that the minimum thickness should be 24 microns, which was introduced in 2002. And the idea was to make them reusable. There were exemptions and exceptions to this particular regulation, and there were attempts to make the thickness minimum thicker, but in fact that has failed because of resistance from the plastic industry and the unions, who felt that the costs of re-equipping factories would be prohibitive and would cost jobs. And so we're stuck at 24 microns currently. And then a plastic bag levy was introduced in South Africa. It's now around 25 South African cents per bag. And again, this was to try and give value to the bags, which has been fairly successful, in fact. Now, very importantly, a new regulation was introduced, and as of the 1st of January this year, 2023, all plastic bags must be made of a minimum of 50% recycled material. This will become 75% in 2025, and by 2027, 100%. So all carrier bags sold and used in this country will have to be fully recycled. So that's a little bit about the national flower. I also want to talk to you about nurdles, which is a relatively new problem. Now, it is indeed a marine pollution problem, but of course, most goods are transported by ship, including the raw material, which are these nurdles, which are high density plastic beads, which are produced and then sent to manufacturers who manufacture plastic products, and they're sent by sea. And there have been two well-known accidents around our coastline. In 2017, this vessel dropped some containers and 49 tons were spilled in the harbor in Durban. And in 2020, 174.5 tons of this material was spilled. And this is a really, really serious problem. And I've got a short video, it's 4 minutes, which I'm going to show you now, which will tell you the story about the nurdles. So let me just quickly open this video for you.
Pushed ashore on every tide, and there's not much anyone can do to stop them. The South African coastline is under attack by billions and billions of plastic pieces. And it's a disaster that has taken everyone by surprise.
How long do you think they're going to be doing this for? How long would you be here for?
Maybe 2 years.
No, not even 2 years. 3
1
years.
year.
Oh,
3 years.
Even 3 or 4.
Wow.
You may not know the name, but the residents of Umtenzini do. They're called nurdles, and they're used in factories to make plastic products. But this group and thousands of other South Africans are now trying to scrape them off the shore. It's an excruciating process.
Once your eye grows accustomed to their shape and size, you start noticing them everywhere. They are everywhere.
A freak storm has torn through areas of KwaZulu-Natal, causing damage to buildings.
It all began 4 months ago when a hurricane wind hit the city of Durban. Streets were flooded and vessels collided in the port, and a couple of containers packed with nurdles fell off this ship. But no one seemed to grasp the significance.
We all work on standard operating procedures. You know what to do when there's oil. You know what to do when there's explosives. Nobody could tell what to do when there's nurdles. We didn't even know that these things behave like this.
10 days after the storm, the government ordered a cleanup. But by that time, it was too late. 49 tons of nurdles had entered the ocean, and an unfamiliar enemy was on the loose.
The difficulty for us is how do we then respond to to such an incident. I do not think that the maritime industry in general has figured out some of the dangers relating to the cargoes that we carry.
The ship owners have hired cleanup teams, and they've been instructed to recover every toxin-absorbing pellet. But it seemed an impossible job to us. We watched this assembly line in Durban try to filter out the twigs and debris. How long would it take to sift through a jar like that?
For 1.5.
1.5 hours to
Yes.
sift through
Yes.
that jar of nurdles. That is extraordinary. So far, they've collected 16 bags of microplastic.
So we haven't even got to the top yet.
And the man in charge told us there's another 40 tons to go.
So we clean up one beach. It looks perfect. We have a spring tide, the sand migrates and blow me down. There they are again. So it's not easy. And I think you can't get disillusioned or despondent. We are recovering product.
That's not something activist Andrew Fenter finds very reassuring. He's deeply worried about the long-term impact on the coast.
From an environmental perspective, this spill is worse than a major oil spill.
It's worse.
It's worse because the plastic's not going to break down.
He took us to a sandbar in the middle of the port that's been officially declared clean, but it was covered in rubbish and translucent pellets.
In practice, when you look at those, you know, they look like eggs. They look like food. And our single biggest concern is that we have sea life, we have bird life eating these.
It is an ecological calamity and a human ordeal, a disaster that's going to stick around. John Sparks, Sky News in Durban.
Right, so that tells you about the nurdle problem. I'm going to move on briefly just to talk a little bit about legislation and regulations. We have a very good National Environmental Management Act, a very broad piece of legislation. And one of the clauses in this act and regulations that stem from it is about the duty of care and remediation of environmental damage. Basically, this law says that every person who might or has or possibly in the future will cause any damage to the environment is liable for that particular harm that they do to the environment, and they have to rectify such pollution or degradation of the environment. And this is the polluter pays principle, which is a very powerful clause. And in fact, those nurdle examples, much of the work for recovering those nurdles is paid for by the shipping companies and the manufacturers of those nurdles. But all regulations, including ones I mentioned earlier, all stem from this particular act. In addition to that, South Africa, of course, is a signatory to several pollution related treaties and conventions, including those that pertain to plastic. I want to also touch briefly on research. In South Africa, there's very active research. We have, as you can see at the bottom, a waste research development and innovation roadmap. This is part of the South African Research Infrastructure Program, and there's an extensive program to address solid waste and waste in general, with plastic pollution obviously being a key element. And then there's a fair amount of academic and technical research on impacts and, of course, solutions, both with academic researchers and by plastic industry players in this field. And then a lot of work by NGOs, both local and international, on the impacts of pollution, terrestrial and marine. I thought I'd just quickly highlight one paper from a colleague of mine called Professor Peter Ryan at the University of Cape Town. And he's done a lot of work on the impacts of plastic pollution on wildlife, in particular on birds and seabirds. And in this, and the point is that the focus this is from the Third International Marine Debris Conference, the focus has shifted from researching impacts to devising solutions. And the point is, we know what our problem is, and we know what the impacts are. What we need to do in terms of research is find a way out of this situation. Okay, moving on, I want to talk now a little bit about the economics of plastics in South Africa. I've given you some facts about how much we produce and how much is recycled. In fact, what's important now is who benefits from plastic. And of course, aside from the attributes we've heard about that we can ascribe to plastics, it also, as a waste product, has value. And in fact, around 52,000 jobs are estimated to be derived from plastic recycling. This is in the informal sector, and it contributes around 2 billion rands annually, saves around 214 or 215,000 tons of carbon by recycling. Interestingly, our recycling levels have been quite stable, as I'll show you in a moment. Looking at the chart on the right-hand side, this is the number of jobs, and in yellow are the number of jobs in the formal recycling sector, but by far the biggest number of jobs are in the informal so-called waste picking or smaller business recycling parts of the industry. So that's what that tells you. The line gives you the tons of per formal employee, tons of recycling that are managed. But this is also a very interesting graph and a chart, and what it shows you in the bars, in the blue is new plastic that is used in the country, which has been fairly stable, but has dropped down over the last few years as people have moved away from plastic. And then you can see the amount, the proportion of recycled plastic that's consumed in the country. As I said, it's estimated that this is worth about 2.3 billion rands. And you'll see on the line that there's this decline in the kilos of plastic consumed per capita in the country, also as plastic alternatives have become more established. Now, the WWF in South Africa estimates that the lifetime cost of plastic produced annually is way, way more than that which is generated from the plastic industry. And so we still have to consider how we can get these to at least equate each other, if not eliminate the use of plastic altogether. All right. I'm getting towards the end now, and I want to just talk a little bit about what's being done. So there's a lot, other than research, a lot of effort in the country. There are lots of different projects, NGOs running projects, such as the water pollution in South African rivers, the African Marine Waste Network, and the Litter Boom Project, which are put on some of the major rivers. And even the industry is organizing around plastic pollution, recognizing how important it is to pay back to society in trying to do this. And they have now established an alliance amongst a whole range of different partners to try and address this problem holistically. So there's a lot happening in civil society. But I also want to say that there's a lot of upcycling that's happening in the country. On the top left are two projects, one making bags and the other making school shoes out of recycled plastic material. So there's business opportunities where people are upcycling by producing material from waste plastic. There, of course, are eco bricks of different kinds. Those are plastic bottles that are used in traditional mud huts to shore up to build what were traditional mud huts, making them more stable and more insular. And of course, there are the traditional eco bricks now as well as experiments to look at resurfacing roads with a plastic a composite product. And then, of course, there are furniture producers. The Adidas project, where they're making shoes from recycled plastic harvested from the oceans. And then Tuftex is a company in South Africa that makes a whole range of different furniture out of recycled plastic. So the important thing here to communicate is that plastic is not waste. It can be used and can be recycled and made into an economically valuable products in our economic system. And then just taking this point to the end is that plastic is not waste. As I said earlier, that about 52,000 jobs or incomes are generated through recycling projects, both formal and informal. And many South Africans, as undignified as it is, are able to generate income. The picture on the bottom left is a very famous picture of Johannesburg so-called surfers, because Johannesburg is quite hilly, and they are well-known plastic recyclers as a feature of Johannesburg. But nevertheless, all over the country, people are harvesting plastic from waste dumps in order to bring them to formal recycling and to generate an income that way. Okay, so I'm going to finish now with a couple of perspectives. The first one is that, as I said earlier, we know what the problem is. We have to both look for alternatives, and where there are none, where we need plastics, we need to really solve the problem of the impacts of waste and use that plastic. I think that in general, consumer pressure is a critical element of any change. And we've seen this in the fishing industry, we're seeing it in climate efforts. It's really the consumers, they're putting pressure at the top end, especially, as I said earlier, the wealthiest people who have the most power to bring this pressure are the greatest producers of plastic waste, can in fact make the changes and effect the changes. It's not going to come without that kind of pressure. So I think that's important. We need to value of waste, and to do that, we need to obviously encourage and incentivize recycling. Legislation and regulation has its place, obviously, and that requires careful monitoring and enforcement. But I would say my last 2 points are my most important ones. Today, we have to address the root causes of poverty, and we have to educate. The problem is that people who live in such grinding poverty as we have in South Africa aren't able to afford the luxury of prioritizing their immediate environment. And so my closing statement is that all people should be wealthy enough to bring effective consumer pressure and have enough in life that the environment they live in can be a priority. And this is the only way I believe we will solve this problem and other related problems. So that's my presentation. I thank you for your time, and I thank you for your attention and the opportunity. And I've finished with the image of the South African cricket emblem, which carries the real national flower of the country, which is the protea. Thank you very much.
Thank you very much, Dr. Navir, for your wonderful. The presentation is covered the whole scenario of the South Africa, what is happening with the plastic waste pollution, and what are the remedies and the regulations your government has been taken over the years. So can you hear our voice? Dr. Navir?
Yes. Hello.
Yes.
Good morning, everybody. I can hear you.
Yeah. So thank you very much again. And now I am opening your talk for the general discussion. So anybody, if you want to ask questions, please raise. Yeah, there's one question.
Neville, good morning. As a wonderful talk, and it has really given us an overview. This is Amitabh from the NAM S&T Center. Can you just briefly outline what kind of comprehensive research program CSIR, DSI, and the universities they are planning over the next 5 to 10 years on this particular problem?
Thank you for the question. Good morning. I can't give you a lot of detail. I can provide you with access to that information, but I should emphasize that the plastic pollution issue here is really integrated with general solid waste management. One of the biggest challenges we have here is, in fact, the collection of waste. Not just the production of waste, but the actual collection and concentration of waste. So there is a significant effort in trying to manage the problem more than just manage the waste production, but also the waste handling and the waste management in general. We have some significant problems of leaching of, in fact, as you might know, in Johannesburg, very famous for its gold mining, and there are gold mine dumps, for example, that have existed for many decades, and these have started to leach, and a lot of chemicals are getting into the water and into the underground water, in particular groundwater. And this has produced a really serious problem for water quality. And of course, incineration of waste exacerbates our already serious air pollution problem. So there are very many different aspects to the research that is ongoing currently, really from all aspects of the value chain, from the production of waste right into the end. So that's a general description of what's going on. Lots of different role players involved, and I'd be very happy to share with you the right information in documentation form. If you're that interested, please be in contact with me, and I'll supply that. Thanks.
So any other questions? Yeah, Professor.
Thank you, Neville. It was a great talk. I was just wondering, do you consider putting on the deposit on beverage bottles, like we do
Sorry,
in Europe?
can you just repeat the question?
So in Norway, we have a deposit on plastic bottles.
Ah.
So you pay when you buy it, and then you get it back when you return the empty bottle.
Yeah. No, there's indeed a deposit on glass bottles, but in fact not on plastic bottles, and I don't know why that is. In fact, I think it you know, to put value on waste certainly is important, especially in a country where there's an opportunity to make money. So if you on refuse collection day outside my house, as an example, every time we put our waste bins outside, there is an army of people that come to sift through that to take out items of value. Any glass bottles get removed, and in fact, so does plastic, because people are paid per kilogram of plastic that they bring to recyclers. But there's no tax or deposit on plastic bottles. There is, as I indicated, on plastic bags. That's more a charge than a deposit, but that does give it value. And as soon as it has value, people treat it differently. So, for example, I never dispose of my plastic bags. In fact, I don't use them if I can avoid them. But if I do, I keep them and reuse them. So I think it's a very important solution, is to add value, even if it's artificial value, by putting a tax or deposit on it. I think that's a very important solution. Thank you.
Good morning, sir. My question is regarding the nodal issue. once the has taken place, or in case of any other climate issue or natural calamities or disasters, we have seen that most of the waste that is produced is of either debris or plastic waste that is being accumulated. So in case of the nodal issue, was there any preventive measures or any extra care taken that was initiated by the government or any other organization? Can you throw some light on that?
Thank you. Thank you. The short answer to your question is no, but the longer answer is that it's actually been quite fascinating. You might know that in June of 2022, there was an enormous storm around the east part of the country in Durban, which caused significant flooding, an extreme event. And this brought the rivers down into the ocean. And I was going to include an image of this in my presentation. I didn't. It is unbelievable to see how much plastic came out of the rivers in an unprecedented way. And I just wanted to say that because you mentioned climate change. Extreme events really exacerbate the problem in the sense that they wash out all of the pollution into the ocean. As you might have picked up in the video, the authorities were really caught off guard. This had never happened before, at least not to our knowledge. And it's been very interesting because there was for an oil spill, there are contingencies. There are booms that are put up, for example, to stop the oil from moving, etc. And for nurdles, they simply were unprepared. Now there is a system in place to try to deal with it, should it happen. But of course, if it happens offshore or outside of the harbor's jurisdiction, which it did happen in the second case, there's nothing that can be done because the stuff gets in the water at sea and then is at the mercy of the currents. And in fact, those nurdles from over 1,000 kilometers away wash up on our shores down in Cape Town, where I live. So in fact, there's been a study published using the nurdle transport in the oceans as drifters to look at ocean currents because they distribute everywhere. So hard to manage, but now there are plans in place. Up until it happened, there were none. Thank you.
Any other questions? Yeah, in the absence of the questions, and finally, I would like to ask you your point of view, because in your presentation, you mentioned that the richest are producing the more pollutions and the poorest are getting earnings from these recycling jobs. How do you think in a way, because finally you mentioned that it is causing a cost for your country. What is your perception regarding this?
Look, it's a complex answer because it's a complex problem. At the end of the day, it is generally the poorer people who suffer the most. I guess, where I'm sitting right now in Cape Town, in a sort of comfortable middle-class home, if I travel 2 or 3 kilometers, I can be in some of the areas with some of the most difficult living circumstances, the highest poverty. And one of the features of a place like that is the distribution of waste, which is not collected from informal settlements. And so really the problem is very complex because of the sanitary conditions in which people are forced to live. And this is because waste is not collected from those areas. So that's one aspect of it. But the other is that, as I said, many people have other priorities. The priority is to get food, to be able to survive, to stay warm, for example, to stay dry in the rainy season. And so the issues of the immediate environment become secondary to survival. And so really, there's that aspect, as well as the fact that the people who produce the waste, mainly those that are wealthier and producing more of it, have more of the responsibility to manage the program, to manage the problem. So, as I say, a complex problem, but at the end of the day, the root cause is poverty in the sense that were people in the position to assert themselves in terms of policy and in terms of their consumer power, I believe that is one way in which we can get a handle on the problem. Thank you.
Excellent, excellent. So with this wonderful discussions, and we can congratulate Sir Dr. Navil for his wonderful presentation with the discussion. So this is a time for presenting the token of appreciation.
Yeah. Thank you, Dr. Neville Swajid. It was a very impressive talk. Now I request our session chair, Dr. Kandaya Pakirathan, to felicitate Dr. Neville Swajid. And in his absence, I request Professor John Sanset, Sir, President, Scope Netherlands, to please collect the token of, as a token of appreciation, the memento.
Thank you very much. Thank you for the opportunity. Thank you very much. Thank you.
Thank you, sir. Now I request Dr. Kandaya Pakirathan to welcome the next speaker, Professor Sharmila Jayasena.
So in the absence of the other two presenters, directly we want to move to the last presenter of this session. So I cordially invite the Professor Sharmila Jayasena from the University of Colombo, Sri Lanka. She's going to deliver a talk on the title of Heterogeneous Microbial Biofilms, a promising solution for combating terrestrial microplastic pollutions. Professor Sharmila, this is over to you.
Check test.
So first I would like to thank NASDAQ, NAM, India, and SCOPE, and JSS AHA, and Sri Lanka, NASDAQ Sri Lanka, and my university for this opportunity to be here. So the topic of my talk is heterogeneous microbial biofilm, promising solution for combating terrestrial microplastic pollution. So this is a dream, an idea that I'm putting forward based on the research I have done so far. So how many of you are wearing plastic today? a few hands showing up. Yeah, so everything I'm wearing today, from my hairband down to my shoes, there is some microplastic fibers in it. So we can't get away from it. It's very hard, it's very disheartening, but that is the truth. And so all these fibers, when we wear it and when we wash it, we let some of it go into the environment. And that is how the environment gets polluted with these microplastics. So these start off as the beads, small beads. Now in the previous talk we heard about the noodles, and they are the seed plastic that the industries use for the manufacture. And yeah, just like South Africa, we had a disaster in 2021, which is considered the worst environmental disaster in our country. So this cargo ship went down in our oceans, and the entire beach and the sea was contaminated. And we were as prepared for it as we were for the tsunami. So it was an absolute disaster. And we had volunteer groups trying to clear the mess a little bit. So, yeah, so when we are tackling this problem, I think we already talked of 4 R's. So there's refuse, reduce, reuse, and recycle. But I think we need another R. So it's not coming. I'm sorry, it's not coming. So the last R was remove. We need to remove the contamination that is already in the terrestrial environment, right? So in that line, microbes we know can metabolize plastics, and they can use it and get the carbon. So maybe this is an area we need to exploit and try to speed up. If we can find a way to speed up the process, then it may help in remediating the environment. So in that line, our research was, we had polythene in our mind. So we looked at alkane degraders. We isolated some organisms. We analyzed the efficiency of degradation. And we also looked at the metabolic pathways of these organisms. So I'll just go over that work. So we isolated, our samples were bits of degraded polythene, which we collected from a municipal dumping site. And we used hexadecane, which is a carbon-16 alkane. It's one of the largest liquid alkanes that you can find. So it's used like a model plastic. So we grew our cultures in that as a sole carbon source. And we were able to isolate organisms that could break it down. So we isolated several bacteria, and we found among them some consortia, bacteria and fungi mixed cultures. And they were actually more efficient than the bacteria alone. We found to be the most simplest consortium and was also very efficient. So we took that for further study. And also you can see in that they are forming a mat. So they were forming what appeared to be a biofilm together. So this is the quantitative analysis we did. So on top, we have the chart for the gas chromatography, mass spectrometry. And what we did was we cultured them either as individual cultures or as the consortium. And after 7 days, we measured what is left in the flask. So you can see in the negative control shows essentially what was there in the crude oil sample. So it had carbons up to carbon 13. And when we grew the bacteria after 7 days, you can see that most of it is still remaining in the flask. So the bacteria on its own was not efficient in degrading the alkanes. But you can see the fungus alone and the consortium, which are the two green ones, there is very little left behind. So we could see that the fungus on its own was also very efficient, but together also they were very efficient. And I don't have the slides here, but when they were together, they were able to degrade it. They were able to start the degradation even faster. So at the end of 7 days, the consortium was able to remove 99% of crude oil when it's in a concentration of 1% in the 20 ml flask. So this was one of the highest reported in literature. So now you might think, okay, if they're so fast, why is still there so much pollution in the environment? One thing is there's a difference between the lab and the liquid that we're using and the actual hard plastic in the environment. And from a metabolic point of view, the first step in the degradation requires oxygen to activate the alkanes. So the alkane hydroxylases or the monoxygenases are not widespread among the organisms. So it's only a few species which would have this required enzyme to begin the degradation. And we also showed that our community, when it's together, formed a biofilm. So these are the scanning electron micrographs. And you can see the organisms are embedded in a matrix, which is secreted by the organisms themselves. And that has advantages for that community. So to say simply, one is it gives okay, so to continue, so it gives mechanical strength to the whole matrix part. So there is all the polymeric substances, proteins, carbohydrates, all that secreted in there. And it forms a barrier for the organisms which are embedded in it. So it stays safe from predators as well as antimicrobials, which is bad from health point of view, but from the organism point of view, it protects them. And also they enjoy an enhanced metabolic capacity because they're living as a community. So there are enzymes secreted, there's increased horizontal gene transfer you find. So if one organism, one species has some enzyme, the other one doesn't, they both enjoy that. So they live in a happy neighborhood. So we could see now that the fungus is able to degrade even the long alkanes, which is in general more difficult even among the microbes. And at the time we knew about the bacterial enzymes, but we didn't know what enzymes were there in the fungi. Although there was a lot of observation of the degradation, they had only identified the CYP52 enzyme in yeast. So we went on to search for the rest of the genes. I assumed that there would be homologs of the bacterial long chain alkane. So LADE and ALMA are long chain alkane degrading genes. So we went searching for these, and we used in silico approach, computational approach. I'm not going to go into the details of that. And we were able to identify in the fungus, which is Aspergillus flavus, we found the CYP as well, and a family of long chain alkane degrading genes, the LADAs, as well as the our maize. So we have published only the LADA still, so don't go and publish the others. So we are yet working on finalizing the work on the other two enzymes. So these are our papers we have published. And so future directions, what I feel is because they naturally form the biofilm. So that has an advantage because it's not a synthetic biofilm. They naturally form it. So, and that gives them stability and a protection. If we could use them to make a formulation, like the way we have pesticides and so on, or fertilizer, if we could use it to, in the soil and maybe even add the biocatalytic enzymes into that mixture, whether that can be used to improve the soil. So that is my team up there, and we work together. And yeah, and thank you. That's it.
Thank you very much, Professor Shamila, for your very influential presentation of remediation of the plastic by using this micros consortium. So now this talk is open for discussion, and if you have any clarification, please.
Thank you. Wonderful presentation and very thorough dealing on this subject. You said that this is for the terrestrial environment. Now, is there a possibility that the same thing can be extended with the combination of this bacteria and the enzymes for any such accident that takes place in the sea? This is number one. So, and
the survival in the marine environment, the fungi are generally, even aspergillus, they say, is very versatile. But we have not tested it yet in that salt level.
Okay. And the
As
second
the biofilm, the bacteria might survive. We are not sure.
Okay. And the because if that can happen in such as suppose an accident has taken place, like Dr. Neville was suggesting, then there is always a possibility of spraying this combination near the port or near the shore and try to see how much degradation takes place.
Yeah.
Okay.
I mean, even if it's not these particular ones, there would be the marine organisms. The same idea can be done, but with other organisms.
Yeah, that requires further work.
Yes.
The second thing is that is it possible to test even in the lab? What is the stability of the biofilm if you have got a saline environment? This is an extension of the other question. So that possibly can be done.
Yes, we can test it here.
Yeah, thank you.
Any other questions?
It was a good presentation, ma'am. I just want to know whether it is a bacterium or the consortium of a bacterium or the fungal organism that you have used in the study along with the enzymes.
What we actually did was our experiments we had all of them in the GCMS I showed. So we had the consortium and the bacteria separately and the fungus separately, just to compare.
Yeah. Which type of microorganism you found effective in bringing about the decomposition
So
or
this,
degradation?
the fungus is Aspergillus flavus and the bacterium was Bacillus cereus.
Any other questions? The professor, I wanted to ask a question, Job. You mentioned that the Aspergillus flavors also, the harmful effects are there too. Yes. So what is your idea regarding this?
Yeah, we actually tested for toxin for this particular strain and it wasn't very significant. But there are issues. Some strains do produce a lot of toxins.
Yeah. Okay. So I wanted to ask you, you have identified the gene responsible for these degradations.
The enzymes.
Yeah. So
Now
is
back
it
to
possible in future to transfer this gene into that one organisms like a gene pyramiding or some other techniques to modify these organisms to increase the effectiveness?
Those are all possible. We have identified the protein, but we have to get at the gene. We haven't isolated the gene yet.
Okay.
So
Okay.
those are the next things we need to do.
Okay. Okay. Thank you. Thank you very much. So in the absence of the questions, and we can congratulate Professor Shamila. This is the time for present the certificate of presentation to her.
Yes, sir. I request Kandaya sir to give the certificate to Professor Sharmila. Thank you, sir. Thank you, madam. Now we are done with the sessions. We shall break for tea for 10 minutes and reassemble for the next session, session 5. And the session chair will be Professor Sharmila Jaisena and the moderator, Dr. Pallavi. And the speakers are Professor Ramamurthy, Mr. Mohammad Reza, and Dr. Sumita. I request all the poster presenters to please display your posters. Please take the help of the volunteers for displaying your posters. Thank you. I request the session chair, Dr. Kandaya Pakiratan, to please collect the certificate. I request Dr. Shivraju to please hand over the certificate to Dr. Kandaya. I request all the poster presenters to please submit your posters to the registration desk. The same thing will be displayed by the volunteers. And I request all the audience to please have a look over the posters that will be displayed. And we can reassemble for the next session. Thank you.
Dear participants, the tea has arrived. I request everyone to have a cup of tea and then join for technical session 6. Very good morning. Thank you for reassembling after the tea break. I am Dr. N. Pallavi, Assistant Professor. So I'll be moderating this technical session 5. And the session chair will be Professor Sharmila Jaishilan, University of Colombo, Sri Lanka. Now I request Professor Sharmila Jaishilan to take her seat as a chairing the session. To introduce about Professor Sharmila Jaishilan. So Professor Sharmila Jaisina is a professor in biochemistry and molecular biology in the Faculty of Medicine, University of Colombo, Sri Lanka. Her research is mainly related to microbial bioremediation of petroleum, hydrocarbon, and microbial biofilms. She has published several research papers related to her research and presented her work at local and international conference. She is the immediate past president of the College of Biochemistry of Sri Lanka. I welcome you, ma'am. Our first keynote presentation is by Professor Praveen C. Ramamurthy, Professor, Department of Material Engineering, Indian Institute of Science, Bengaluru. The sir will be giving a talk on microplastic challenge, molecular solid state sensor. Now I request Professor Sharmila Jaishin to please introduce our keynote speaker.
Thank you. So welcome to session 5. So we have one keynote and 2 other speakers for this session. So our keynote speaker is Professor Praveen Ramamurthy from the Department of Materials Engineering, Indian Institute of Science, Bangalore, India. Professor Praveen received an MSc in Polymer Science from the University of Mysore and a PhD from Clemson University in 2004. He worked as a R&D engineer in Thermodisc, Ohio, USA, and as a research scientist in Hoku Scientific, Honolulu, Hawaii. He joined Indian Institute of Science in the year 2007 and is currently a professor in the Department of Materials Engineering. His current research focuses on organic electronics and sensors and biosensors. He has over 270 peer-reviewed papers published and 21 patents to his credit, 18 PhD and 20 masters, and 8 undergraduates have graduated from his group. So welcome, Professor Ramamurthy.
Thank you. Okay. First of all, thanks for inviting me, Professor Suryanarayan. It's the first time me coming here to this campus. Thank you very much. As I said, we work on organic electronics. We work on design. Am I too loud? It's okay. It's okay. Design of molecules to tailor its properties. Right? That's what we do. What do I do the connection to microplastics or plastic pollution? Right? That's where I am taking further with this. So we design molecule, change, alter, tailor its electronic properties to push a particular characteristics to come to the fore. Something else will become predominant. That's what we do. So for that, let me show a few work that has happened on how do you push this particular properties. Okay, and then I will come to the microplastic part. Yeah, I'm from IISc. This is, I just wanted to show few nice looking photos of the institute. This is our campus right in the center of Bangalore. If people have not come, please do come. I'm sure some of you have visited this. This is how it used to look 100 years ago, and this is how it looks now. So with that, I'll start. Okay, so as I said, we do make. Okay, I think the red laser doesn't reflect on the LCD screen. That's okay. So anyhow, so we have, as I said, we make various molecules that interacts with various different analytes. Just for the better way of explaining, I am showing 3 or 4 different kinds of analytes. For example, metal ions, volatile organic compounds, right? It's an organic material, or nitrate, a inorganic complex, or some biological organisms. All 4 are entirely different kinds of material. How do I make certain molecules to interact with that, right? That's what we work on. Very quickly, I'll go through this so that it puts a better perspective in how we are going to address the microplastic part. Okay, here, as I said, the number of analytes that you detect is limitless almost. I mean, I can have, you name an analyte, whatever the form it might be, you could create, the chemistry is such that you can architect a molecule that interacts specifically with that. So example here is, we can detect anything. That's where the last part of this, it comes here, is microplastic, right? So can you design something that interact with this plastic so that not just some spectroscopic technique or some inductive coupled plasma or something like that you detect, can I detect by solid state? That's the idea with these sensors. Okay, so what do? This is what we do. We make molecules, we design molecules. If you look at these molecules, I mean, these are the examples of molecules, the top right and bottom left. Looks ugly looking. Basically, what you do is, I mean, we don't have too much play with this. I only have carbon, hydrogen, and then you have oxygen, nitrogen, sulfur, phosphorus, and things like that. So you architect the molecule such that you have push and pull mechanism. So you push and pull the electronic energy levels such that you change the energy levels. Not just the band gap, you also change its conduction band, valence band, HOMO, LUMO, in particular set of energy levels too. If you look at it, the molecules that we make, the top left-hand corner, you have band gap of 1.2 to 2.6. This is all organic molecules. So you architect such that it changes. Not just the band gap. If you look at in the bottom center part, you have that red dots all over. So it's basically, the top one is conduction band and this is valence band. You can see by designing that molecules, I can change these two, not just the band gap, alignment of these two. Why I am telling 100 times the same thing is, this is so important. The designing part of a molecule to change the electronic properties. So you make, that's the design part. Then you make a device. What device? So you have so many different types of devices, right? So basically, the idea here is to make a solid state device. It need not be, as I said, electrochemical or spectroscopic. So I should have a field effect transistor, FET, or a resistive, a capacitive based sensor. I take it, make it interact with an analyte and look at its signal. That's very easy, right? So you make various different architectures. I'll just take a couple of examples and show you here. This is one of the the device that was made some 6-7 years ago in our lab, where this, what you see here is the sensor. That black color is the molecule that was synthesized. Very simple resistive based sensor. So the students at that time rigged up a Arduino, Raspberry Pis, and then just look at the signal, and then your cell phone has so much computational capability. Push it to cell phone, and then cell phone can key in its calibration values and interference, pattern recognition. All of this can be made, and then you can actually get some, just by reading a resistance, you can get what ppm level of that material is, right? That's how the thought process is. And that's what we did actually, right? Here, the example we did was the molecule, for example, nitrate. Nitrate is a major challenge. So if it is there in the water, I take this resistance, put it in, and then my cell phone should tell me how much nitrate is there. So now I can have a GPS location. So I know map. I don't have to go everywhere, take samples, go to each location, measure it, and then you can create a map of that particular pollution, right? A particular analyte here. Okay. Nitrate, let's come to the nitrate. Nitrate is, what's the problem? Nitrate is not a major problem. It doesn't kill you overnight. but it's cumulative. There's a huge problem because of a lot of fertilizers that are used, and it's never used by the plants, right? Majority of it that goes down the drain or the rain with the rain, and then it comes back to our whatever drinking water. So how do you detect? So we do a lot of computational chemistry, design a molecule that interacts specifically with nitrate here. And if you look at it, for example, we do simulation after understanding that interaction. For example, if there is a HOMO to LUMO +1 interaction that happens at 397, that is happening only by the nitrate and our analyte. Okay, so and if you look at it, that's what it looks like, right? So mechanistically and theoretically, you prove there is an interaction, and hence that's a sensor. So if you look at this, again, sorry, because of no other way of explaining, I have to show you some data. Don't worry about the numbers, but just the concept is what I would like you to take it further. So if you look at it, this is a response curve, response data. This is for nitrate and everything else, and the nitrate is in the topmost. So that is statistically different, and this is some 2000 to 2500 samples, number of samples over 3 years of time, right? That's a lot of data, and it's statistically sure that you have your molecule interacting specifically with the nitrate. All you did was you take this a solid state device, put it inside the water which contain, and then you have this, right? And also it is very repeatable. If you look at it, you take it in DI water, put it in some 10 ppm of nitrate, the response goes up, put it in DI water, it comes back. That means decoupling happens. Again, put it in some constant. So you can keep on doing this. So it's very reversible, right? That's also very important for a device. It cannot be use and throw, right? Another one. Okay, I'll just, as I said, just few more examples before I go into microplastics. This is another example of very busy slide. You look at the top left-hand corner image. That's a spectra of the polymer that was made that interacts specifically with zinc, right? Zinc, if you look at any other element that is there, it's not interacting, right? There is no change in the signal. With zinc, there is a huge exponential increase in the signal. So that means this guy interacts specifically with zinc. Zinc, where is the problem? Zinc, basically, you don't again go search for zinc. What is the problem? Where is the problem? You only, if people with dementia, if there is some imbalance in the brain, you would look at, go back and the doctor will go look at, measure spectroscopically how much zinc content is there in the blood, right? Otherwise, you don't do that. So the idea here was, can we do it by like a blood glucose measurement, right? So you make a small device, a small drop of blood, put it in. This is an FET structure, field effect transistor structure, and you can detect as low as 1 ppb, right? That's instantaneous, right? Again, I'm just trying to build up the case how important to have a solid state sensor, right? That's the idea. And very specific for that matter too. Okay, I'll move on. The next one is E. coli. I absolutely don't know what an E. coli is, but I know E. coli is something that should not be near my food. My food should not come in contact with that. That's all I know. But E. coli is an organism. Every organism has its skin. Every skin has its own specific functional groups that are predominant. All skin are same, right? But some functional groups are predominant in that particular skin. So you again go back to chemistry, look at that functional groups, design a molecule that interact with that specific group, and then hope that you have a device that detects this E. coli, right? So again, I'll not go through, but the point is you made a molecule that electronic energy levels are similar. If you, this is a simple device you made. If you look at this top right-hand corner, this is inductance and this is resistance measurement. Inductance measurement, I can measure 1 CFU per ml of E. coli instantaneously. Right now, E. coli measurement takes, some of you might know, it would take 24 hours to 48 hours because you need to culture it, incubate, grow, and then look at through microscope and say, look, there is E. coli, and then hence I will say E. coli. But if I have a device that I take it, put it there, and then say there is a specific interaction, there is a change in signal, that's wonderful. That's what we've been working on. So, and also it's 1 CFU per ml. That's almost unheard of things. Okay. Next, what we are trying to do is, can this also be interaction is happening? There is a signal. That interaction, can it also make it as a filter? Right. That's the idea. Okay. I'll move on. Last but one, I think, is urea. Urea sensor. Urea. Where is urea from? One, of course, fertilizer. There's another problem is urea adulteration happen in milk, right? That's the biggest problem. You can't measure it, right? Anywhere in the field, how do you measure a quality of milk, right? Milk you measure by its density, right? That's the easiest. But I can play around with density. I put some oil, put some urea, I can bring back the density, put some water, and then so I get, instead of 1 liter, I can have 10 liters. And it is known people do that, just any geographic location, many places, right? So how do I detect? Again, I have to take a spectroscopic instrument, which is huge, somebody trained, or can I have a device that measures this? So again, here we made some molecules that interacts. If you look at this, it's almost statistically so much different than anything else present in milk, right? So again, the example is, you have a molecule, you have a device that specifically interacts with urea and tell you there is urea, how much urea is there. Okay, I'll stop with this, saying that you can bring out any element saying that I need to detect this, we should be able to detect. So example here is copper, lead, iron, chromium, fluoride, name it. And the examples I shown you of biological organisms, metal ions, inorganic complexes, and things like that. So if I can do all these things, can we do the same thing for microplastic? Okay, there are only 2 examples I'll take. I'll finish if time is up. Please let me know. So one of this is polystyrene. Okay, polystyrene. I'm sure many of you here in the audience would be working on various different microplastic remediation and things like that. One of this is polystyrene. Polystyrene, there are enough. If you Google Scholar Alert, do this, you can find n number of articles that says polystyrene in the blood or fetus or some food particles, anywhere you take about. Idea here again is, how do I see, how do I detect that? There are many methods that you detect, right? Whereas a lot of them are very challenging, calibration challenge, sample challenge, and also detection limit and interference, various different. The thought process that happened for other sensors, can I make a molecule that interacts specifically with polystyrene? Okay, so we started doing computational chemistry. Again, design, basically you do literally 100 molecules before you come up with some 10 of them that can interact electronically, right? It should interact in many different ways. One of them is electronic, second one is size, third one is shape. Size and shape is matters with other analytes, but plastics, the fragment, if it is a monomer, that is okay, but it's a fragment, so size and shape goes for a sixer, right? You can't catch that. So most important is its electronic energy levels and charge distribution, how much positive charge and how much negative charge, and how it is distributed on the backbone indicates how well it can connect. For example, here I said polystyrene, and there's a molecule that was designed similarly by other method. We showed that there is a pi-pi interaction happens, perfect match with a polystyrene molecule. There is at particular pH, that's still a challenge, but at particular pH, if you look at it here, both of them has similar energy levels. This is basically to elucidate whether your molecule can interact or not. Okay, that's what you do. Skip. So you made to match the analyte size. Actually, the student tried, instead of me having that molecule itself, can I make a microspheres of this so that at least the size of these both matches? So that's what we did. Once that happens, this is analysis of that molecule, whether we got it. Chemically, it is the same structure, and physically, it is the same size and shape and things like that at molecular scale. This is not at macro scale, right? We how did we? So we added with the polyester. Here, it's difficult to see, but I'll come back to the next few slides. But if you look at here, the red one and the black one is where your polystyrene was. And when you added this molecule, okay, for some reason they put it as PDA. It's some nomenclature of the student. But anyhow, so if you look at that, the blue one, the absorption has come down. So that means basically what happened, the polystyrene which was there before is not there. What happened to it? The molecule that was put interacted electronically and then brought it down. So in addition to detecting that molecule, also it coagulated and trying to separate, right? That's the idea, doing two things at the same time. So I will show you a close-up image of this. So we did a lot of different concentration of this microplastic and see whether we can both detect and separate optically, right? Still in the optical stage. So yeah, I'll again not go through the numbers here. That's what has happened. You proved that it has happened. I'll skip. And I mean, any sensor you do, you would never do one or two sensors, right? Some of them work, some of them might not work. I could not run with that data and then go publish. Publishing is still possible, but that doesn't make sense. You would normally do a statistical analysis for this. So you need to have n number of samples. So statistically, it should tell me that that is happening. So that's what happens here. And if you look at this, each one has more than 200 number of sensors that was used. You could at different loading, different pH for various long duration and interference. If I, instead of polystyrene, if I put something else in that, any other element, does it even still interact or it doesn't interact? So these are the data. That data says it's very specific. This guy only interact with polystyrene and I can detect, right? That's the idea. And detect, in addition to that, I do something else with the molecule, it coagulates and separates also. So it's basically cleans up. It tells me there was polystyrene and now I cleaned it up. Okay, that's what it does. More statistical data, and I will not bore. If somebody is interested, at the end of the talk we can discuss. Basically, all of this says particular condition I can detect optically right now, not conducting method, but optically I can detect and I can separate. All right. On different condition, different things. This is a good spectra, and you can see with blue one, with the addition of that sensing material, it just changes the optical property, right? That means interacting and then separating. Okay, I'll stop this here. Let's go to the last one, is BPA. BPA is also another challenge, whether it is a fragmented portion or just the BPA itself. BPA, everybody, if you don't know also what a BPA, bisphenol A, at least you would have heard in the news items that BPA free is your bottle, use BPA free. Basically, it's a polyester polycarbonate based material. When it breaks down, that bisphenol A will, like, it's supposed to be carcinogenic and things like that. But can you detect that? That's another, again, whether it's a fragment or at the molecular scale, it doesn't matter. But can we detect, right, solid state? Hence, again, we did a lot of molecular modeling. Look at two things. In one more addition was done, right? Last couple of years. What is called metal organic frameworks. The metal organic frameworks are there from long time, but last few years it is coming back into trend, right? Everybody looks at MOF and COFs and MOFs are looked at, and because it's a cage structure and you can change the element and change the cage size. That's fantastic. That's what you need for something to hold on to, right? So you design some COF and MOF and also some molecule that electronically interact. So you have interaction and you have something caging, right? That's what this was done. So this, in addition to that, if you look at this molecule, it is conjugated. So conjugated means alternate double bonds, means alternate pi bonds. Alternate pi bonds means one is sigma, other is a pi bond, other pi bond is loosely bound electron that can break and move. So if an electron cloud can move from one place to another, that means it's become conducting. That's how it's become a conducting polymer. So because of this interaction, my pi bond breaks and then you have charges moving. So that means I can detect, if I detect that movement of electrons, and then I can detect there is an event that is taking place of interaction of this molecule with the analyte, right? That was the idea. So you made electrochemical sensor with these composites and things like that. They also have done a solid AFET based sensor on this also. So if you look at this at various different concentrations, with various different configurations of electrodes, you can detect, right? That's again was unheard of things, unless you do an spectroscopic technique. So you have something, I take it, put it in, I know whether BPA is there or not, right? That's the very neat part of it. Again, you look at various interference, you can see arsenic, barium, cadmium, iron, mercury, potassium, nickel, you put anything with that, still this molecule interact very specifically with BPA, and then I can measure that, okay? And it's so stable, stability, repeatability, reproducibility, interference, these are important things for a sensor. Okay, with that, I think I will stop. There are data that showed how effective it is, how I can not just detect, I'll extract it out and things like that. I'll skip. I'll stop with this. So last couple of years, I mean, we have been working on sensors for so long. So institute encourages faculty to do a startup kind of thing. So we have started a company. Now the sensors as small as a pen, where I can go keep it in this. The front portion is the sensor, which is you can remove exactly like your blood glucose monitoring system. So you put any sensor, whatever analyte you want to measure, put it in, go there, and it will be uploaded onto a IoT based system. So you can create like a Google map, you can create a map of a particular analyte, right? Whatever many analytes which I showed about. So with that, we are hopefully we are there, and I've been telling last 6 months, in next 3 months I should have a product ready. But making a product is a different beast, making a molecule in the lab. So, but that's where we are in. With that, I'll stop. If there are any questions, please let me know. Thank you for listening.
Thank you, Dr. Ramamurthy. That was a very interesting talk. If there are any questions? So,
the first question is in regard to arsenic detection. I'm sorry. See, arsenic, the WHO limit is 10 ppb. Now, earlier those field-based test kits that was there. If you put see the development of color, this only gives you a range.
Correct.
It doesn't, depending on the shade of the yellow or shade of the brown you want, you make a guesstimate that. So for arsenic, suppose if you are considering say 5 ppb to 25 ppb, let's say. So in this case, what is the kind of accuracy you get in this kind with this kind of sensor?
See, this interaction is electronic.
Yeah.
So quantization of that interaction depends on how good your electronics is. So if you have a good electronics, we can measure as low as few ppbs. You saw in zinc, we can measure 1.6 ppb, and that is the error bar also for that is less than 1 ppb. So I can measure that. But keeping in mind the biggest problem in sensor is false positives and false negatives. Even if it is there little bit, it gives me an alarm, is not a good thing. So hence, this chemistry is important. Looking what is your WHO guidelines or whatever is the requirement, the trigger point should be around that. You said 1 ppb.
No, it is 10 ppb.
10 ppb.
It should be below 10 ppb for drinking water.
Correct. So if your resolution is less than 1 ppb, then it is a good sensor.
Absolutely.
So hence, so all the work that we do, molecule design such that it should be in that range.
Okay, that's fine.
So it is less than a ppb.
Just a second question about the molecular architecturing. The kind of model you use, they are deterministic or a population balance kind of model?
Oh, I am not a simulation molecular guy. Students who does, I don't understand literally half of it. But they use both the DFT based calculations and also material studio molecular modeling, dynamics kind of thing.
Okay, sure. Thank you so much.
Yeah. Any more questions?
It was a very good presentation, sir. My question is, you made some molecule react with the E. coli bacterium. Like E. coli bacteria, as per the cell wall construction, it is an example for gram-negative. So whatever binding, whatever interaction study that you do for E. coli, will it suit for gram-positive bacteria as well? There are so many staphylococcus stuff, like streptococcus.
I am not, as I said, we're not going to that macro scale. Either gram positive or gram negative is not I'm looking at. I'm looking even lower, one more level lower, where I pick up some of the functional groups on this. So if I make it as equivalent as any gram positive you do, I get a lot of false positives. Interference will come. So hence you go in another level, look at that particular functional groups, and it has to only interact with that. So it won't be a general any. So the next level we would like to work, we want to start working on is what strand of E. coli. Not all strands are, again, I'm not a biologist or a microbiologist, but some strands are lethal and some strands are okay. I mean, all of us have E. coli. Nothing. So that is the work that is going towards. Okay, there's no more questions. Thank you.
Okay, actually, I just wanted, so
Yes.
you make a polymer and it's applied onto a surface, is it?
Sorry?
Is it applied onto a solid solid for the device?
Huh.
So you construct some kind of polymer, and then it is it on, like you lay it on some solid surface to get?
Depends on the type of device. If it is a FET, if it is a resistive or a capacitive based, you just coat it over it. But if it is a FET where it is the active layer is embedded in, then you actually grow, right? So sometimes we take the FET architecture, bare architecture with a gate and a drain, then grow the polymer on it, and then go put another electrode. So it depends on what type of electrodes device you are making.
So I think it's a gold mine that you have here, because as you say, you can detect anything.
Right, right.
Right. So yeah. Congratulations.
Thank you.
Thank you, sir. Now I request Dr. Shivraju HP, coordinator of this international workshop, to give a token of appreciation.
So we will move on now. The next speaker is Mr. Muhammad Reza from Indonesia, who will speak on the spatiotemporal variation and seasonal dynamics of stranded beach anthropogenic debris on Indonesian beach from the results of nationwide monitoring.
Thank you.
I request the speaker to be
So it's
strictly
one
follow the time limit. So it's
back.
12 minutes plus 3 minutes for discussion.
Good afternoon, everyone. So I would like first thanks to the sponsor, to the NMNST, GSS, and also Scope for inviting me here. It's really big honor for me. To be honest, I'm quite surprised that I'm invited here because I'm particularly working in the marine area, not in the terrestrial one. So it's kind of a quite, nervous right now. And also it's almost 3 years that I'm just giving talk in the front of the monitor right now and in front of the really expert in here. Hopefully everything is going to be fine. So today I will present everything about the stranded beach litter as I working in the research center for oceanography. So I prefer to call myself not a researcher, sea and also in the beach. So once again, thank you very much. My name is Muhammad Reza Cordova, but you can call me Reza. I'm from Research Center for Oceanography, the Indonesian National Research and Innovation Agencies. So Indonesia, you already knew that it's located in the Southeast Asia. It's home for the marine tropical ecosystem, like it's the center for the coral reef, center for the mangrove area, center for the seagrass area. It's support more than 270 million people who live indirectly and directly into the ocean. So, but the problem is there were so many pollution that affected by the Indonesian anthropogenic activity. One of them is plastic. Maybe you already noticed this morning and also yesterday, Indonesia is considered as the top 10 polluter of the plastic pollution into the ocean. And most of them are coming from the river. So this is waste management that happened in Indonesia until today. So we have like a big pile, like a mountain in here. So we just put it like the open dumping, just bring it all the waste from the household, from the office, from every the collection rate in Indonesia is not really good. It's only 56% of the area that's collect. I mean the waste is collected, and that's caused something like this one. This one is the footage video that I get from my colleague in Sungai Watch. It's the one of the NGOs that's based in Bali. You may see this is an illegal dumping area that is really located just next to the river. And then you can imagine if there's any, rainfall, and then somebody will, or maybe the wind, it will bring all the waste from here into the river. And of course, it will end up in the ocean. So based from this, UNEP said that most of the marine plastic litter or marine plastic waste will go to the ocean. So 80% of the plastic waste that produced by the land-based activity will go to the ocean. 20% of it is coming from the sea-based activities. And then with that reason, we have a 3 research questions that we have to answer. The first one is how much the marine plastic litter and which area are affected the most, and then where does it come from and how do we tackle it? And then what we did is we do the monitoring in the stranded beach litter. So why we did this? Because it's kind of a really easy, not really give money too much to the volunteer and everything, and then we kind of adapt the method that already established by NOAA or NOAA and also UNEP shoreline debris assessment. And then after that, we did the sampling in 18 area in Indonesia, 9 area in Western Indonesia, and 9 area in the Eastern area in Indonesia. You may see from the red dots, it's the Western part, and then yellow one, it's on the Eastern part. And then after that, we train volunteer to have the same vision with them, and we have the same method. And then what we did is, before we do the sampling on the month zero, we clean it the area first, and then we kind of identifying what kind of a waste that we found in the beach. And then after that, we collected monthly every time. So every volunteer did the same at the exact time, location, and at also similar time during the spring tide or full moon or new moon. And then this is the result. The first one, of course, the plastic and rubber is the dominant waste that we found in the beach. It's almost 50% of the waste is plastic, and it's contain more than one item per meter square, and also more than 80 grams per meter square also. And then with this slide, with this figure, you can see on the graph A, it's the location. There's a big variance in each area. But the interesting in here is you may see in the picture in the middle, the waste have a correlation with the season, have a correlation with the rainfall. The higher rainfall, it's caused the higher waste that we found in the beach. And you may see also in the rainy season on the C figure, it's have a really high and have a what's it called? The significant difference of the waste that we found in the beach. And then it might be clearer because in each area, we can see the number of the plastic are really increased during the rainy season between the November until December in terms of abundance and also in terms of weight. It's quite interesting, but most of them we already knew that, right? Because it's all the waste are brought by the water. And then the other interesting thing is we found more than 70% of the waste. Oh, sorry, it's not more. It's almost 70% of the waste that we found is single-use plastic. The most single-use plastic one that we found is the plastic multi-layer sachet, and then the second one is the plastic bag, and then the plastic bottle. The interesting thing is the farther you are sampling from the Java Island, which is the center of Indonesia anthropogenic activity and also the center for economic activity, the more you find the recyclable plastic. For example, like mineral plastic bottle or mineral plastic cups, they common in outside of Java Island. Why? We think we suspect it's because the recycling industry are more centered in the Java Island. Outside of Java, there is no recycling area. So that's why we argue to and give information to the government that we have to invest more into the recycling industry outside of Java Island. If we compare the beach stranded litter in whole around the world, but it's not all, I just put some of them. If you see the green one and also the red one, the green one, that means it's lower than Indonesia, and then the red one is higher than in Indonesia. If we compare, like in Indonesia, it's higher than in Philippines, in India, in Kenya, in Brazil, in Morocco, in Norway, and also in US. But if we compare between in Sri Lanka, in South Africa, in Turkey, and also in Spain, the number of stranded plastic debris is much lower. And then we also did the preliminary modeling. It's coming, the stranded plastic debris that's coming, it's coming from the local area, like from sea top right, tablet, where does the stranded beach litter that's stranded in the north of Java? It's coming mostly coming from the local area. And then when the river release the waste from the north of Java river, it will go also stranded in the local areas. But the bad news is some of the waste are going and end up in the Indian Ocean. I will talk more about that. And then the other modeling that we developed last year. This is the forward trajectory of the riverine litter that come from 10 major rivers in Java and Bali, and most of them, of course, will end up in Indian Ocean. And then what we found in our research was more than 60% of the stranded beach litter is a fresh waste. As you may see, there is no, something inside, just a small level of biofilm, and only 37% is all waste. You may see on the right side, there is a like a kind of a barnacle, but it's really not common in stranded beach litter that we found during our sampling time. And then we can say that 60% of the stranded beach litter, it's come from local source because it's fresh waste. And we think it's also predominantly from the river, particularly in the local areas, not really far from the stranded beach areas that we. So this is the other modeling that we got from our colleague in South Africa. So they did the sampling in Seychelles Island. So most of the countries in the Indian Ocean that will cause the accumulation of the plastic debris in this area. So you can imagine now if there are so many plastic, so many waste that are transfer, or we call it the transboundary debris that are coming from one area to the other area. So this one is, we have to tackle this because most of the debris is coming from the land-based activities. So what should we do? This is kind of a simple and effective. We have to put at least the river trash boom. So this one is, for example, on the top left, top left. It's the river trash boom that we installed in Jakarta. This is the picture when during the COVID-19 on the end of 2020. You can see we found the hazardous material because most of the people just threw it, in the environment. And then Sungai, more than 10,000 barriers, if I'm not forgotten. And there are also river trash boom that installed in South Africa, also in Australia, and also in Malaysia, if I'm not forgotten. We have to talk more about that. And then we also already gave our recommendation to our government to install the river trash boom. So we made a calculation, at least if we put or install the river trash boom, and also we also invest into the like the waste bank or any better good management, we estimate the number of the plastic can it can reduce until 50% in 1 year. So maybe you already remember that Indonesia have a target for really ambitious target. So our government wants to reduce 70% of the plastic waste that goes to the ocean by 70% by 2025. Maybe we can do something like this, and then we can tackle it from the source. Not really from the source, but it's kind of like a fast or emergency strategy that we should do. So if we can say that the tackle of the marine plastic litter in the river can minimize the plastic litter that goes to the ocean, and of course that will minimize also the transboundary litter, because I'm very, very sad if I saw some product that maybe is stranded in India or stranded in South Africa. Based from our model, we just afraid we don't want to pollute the other area outside of our countries. So as a take-home message, and also for the conclusion, the first one is, of course, the plastic dominated stranded beach litter in the anthropogenic debris in Indonesian beach, also similar with the other countries. And the stranded beach litter is correlated with the rainfall and seasonal. And there's a source of the stranded beach litter is mostly coming from the local areas. And then we have to tackle by the source, it means in the river, because we have to be aware about the transboundary litter. So I think this is the reference, and that is coming from me. Thank you for listening to my presentation. Thank you very much. Hopefully you enjoyed. Thank you very much.
Thank you, Rezi. The floor is open for questions.
Thank you, Dr. Raja. You have given a very nice presentation. I have one query. As you told, seasonal effect due to rainfall, surface runoff, plastic waste get accumulated somewhere in river body.
Yeah.
But how we can say this is a effect of seasonal or a special temporal variation? There is no temporal variation.
Exactly.
So just elaborate, just my query.
Well, that's right, because we now working with the other research project. After we did this research, we just realized because like the habit of the people, mostly they consume the plastic quite a lot, and then they just do the litter. They just littering every time they want, right? Like every day. During the dry season or during the transitional season, they also just do the littering. For example, like do the littering, and the waste is accumulated in the river bank. And when the rain come, it will wash every waste that already accumulated in the riverbank. And that's why we think there is a higher probability of the waste that's stranded during the rainy season because of that. So that's why we kind of have not only tackle the waste during the rainy season, but in all time. So that's why I kind of agree when yesterday the keynote speaker, because I saw there is a picture showing Indonesia already also make the plastic tar road. I also agree with that. And we have to do something like that. But I do agree with you. It's not correlated directly if we think about the activity in the land. But for the plastic release, it's related because the rain, the water in the river will just wash every accumulated waste that already in the riverbank. Hope that answers the question.
Thank you, Dr. Rida. It's a very interesting subject.
Thank you very much.
I'm wondering if you find some variation between weekdays, weekends, and variation between districts, poor areas, rich areas, because I think culture also could affect the distribution of plastics. Have you find something like this?
Oh, that's a good question. To be honest, we haven't done that research yet. If we can do the sampling like a daily or weekly, it's also our target before we did our research, but due to the limited budget, unfortunately, we can only do in monthly. But in the future, we plan to doing that. One of my PhD students have a proposal to doing that like every day. We'll see. Hopefully, we got the funding, and then I can give you the answer after we publish the result. Thank you.
Thank you.
Are there any more questions? So in the absence of any further questions, thank you very much.
Thank you very much, Professor.
So we have a certificate to give you.
Should I take the elephant? Very much. Thank you very much. Thank you very much, everyone.
Meanwhile, I would like to announce that the resolution was given yesterday to all the participants or the delegates from us. So I request you to give your valuable feedback in that resolution and return back during the poster session. After this session, I request you to return back the resolution which was given with any additions or changes which you want to include in that resolution, which we have come across of these 2 days international workshop. Thank you.
So we will move on. The next speaker is Ms. Najiha Bindi Mohammed from the Fisheries Research Institute, Jalan Batu Maung, Malaysia. She will talk on the distribution of microplastics in water, sediment, and mussels of Langkawi Island, Malaysia.
So namaste and good afternoon to honorable
speakers, moderator. My name is Najiha Muhammad. I'm a research officer from Fisheries Research Institute, Department of Fisheries, Malaysia. And today I'll be sharing about my findings, distribution of microplastics in water, sediment, and mussels of Langkawi Island, Malaysia. So basically, I'll be dividing my contents of presentation into 4 different sections. That is introduction, methodology, results, and conclusion. So for the introduction, I won't be going very detailed of what are microplastics, as most of us have already learned and knew how microplastics came from. But I just wanted to emphasize that Malaysia has been ranked 8 out of 192 countries in mismanagement of plastic waste, with estimated amount of marine debris 0.14 to 0.37 million metric ton per year. And because of this mismanagement of waste ends up in marine debris, and due to constant exposure to light, to UV light and low temperature, causing plastics to turn into microplastics. And the evidence of microplastics in marine biota there have been many evidence of microplastics in marine biota. And according to Consoli, the ingestion may occur because of chasing a small prey, or probably because of accidental ingestion. They may also be mistaken as food because of their small fragments. And there are evidence from a state in Malaysia that found 149 to 500 micrometer sizes of microplastics in GIT and gills. And apart from that, microplastics are not the only concern, as microplastics are acting as a vector of pollutants. As studied mentioned, the crab exposed to microfibers experience decreased reproductive success and severe stunted growth. And apart from that, they're able to accumulate chemicals like carcinogenic chemicals like PAH, PCBs, and PVDEs. So our objective is to quantify the distribution of microplastics in seawater, beach sediment, and marine mussels of Pulau Langkawi, and to investigate the relationship between microplastics distribution according to temporal. And our hypothesis is the distribution of microplastics in urban and suburban areas will be higher because of population density and human activities, and a higher occurrence of single plastic use sources. So for the location, actually our study focused on the Straits of Malacca, focused on the Kedah coastal waters. But for this presentation, I'm only sharing on the Pulau Langkawi. Pulau Langkawi is considered a high tourist spot, not only among locals, but also in foreigners. And they have a lot of industries, not only recreational industry, but also fishing industries. And they're boasting about 94.7% urbanization rate. And they are also listed as the only geopark listed, the only UNESCO geopark listed in Southeast Asia. So considering these factors, we chose Pulau Langkawi as our location. So this is the methodology for our study. Seawater samples were collected using a manta net trolled at the back of the boat for 10 to 15 minutes at a 2-speed node. And for sediment, we conducted transect sampling where the high tide line, mid tide line, and low tide line is collected and then transferred into aluminum foil. Then for the marine mussels, we obtained them from fishermen. So all of these samples were later taken to the laboratory for further analysis, where we adapted NOAA for seawater and sediment. It involves sieving, drying, wet peroxide oxidation, and density separation. While for biota, we use 10% potassium hydroxide for digestion and then filter. So all of the samples are later categorized according to shapes, colors, and sizes. And for further understanding of its resources, we conducted it using micro FTIR. So basically, this is the results for microplastics concentration in seawater surface. The total concentration for all months on all stations is 16.20 particles per meter cube. I didn't mention before, but ST1 is actually considered a private pilot access to the area, while ST2 and ST3 boasted many industries, like they have recreational activities, fishing industries, aquaculture industries, and ST4 is near to residential area. So contrary to what Mr. Reza presented before, our initial Our initial thought was that microplastics concentration will be higher in rainy season, that is in September, November, but the result is otherwise. Microplastics are found in certain stations to be higher in May and July, and fibers found in all samples with 45% followed by fragments and foam. And microplastics concentration differs significantly between stations. And this study is slightly higher than the east coast of Peninsular Malaysia, but is still lower than the Gulf of Thailand. So we actually found 19 types of polymer from this study, and we found rayon that usually comes from textiles as the highest source of polymer, polystyrene, fluorocarbon polymer, polyvinyl alcohol, POM, MF, so many different sources of polymers. And the rayon is similar to local studies and also globally. So this is the results of microplastics concentration in beach sediment. For SS4, we are unable to access the location, so that's why we only have 3 locations. And SS2 recorded the highest average microplastics concentration in May, July, November with 460, 740, and 760 particles per kilogram. And SS1 recorded the lowest average concentration for each sampling month, which is similar to the results found in surface water. I was told by the hotel management, SS1, actually, they conducted frequent beach cleaning around the area. That's why explains the low concentration for each sampling month. And fragments were found in all samples, foam, fibers, and beads, and significant difference was detected between microplastics concentration to sampling month and stations. So we actually found 14 types of polymer, but for this purpose, I only shared a few, as the other types of polymers only boasted about 2%. So we found polyethylenimine or PEI as the higher source of polymer. PVC are usually found in most other studies, but PEI are not usually found in most other studies. To date, I only found one from Italy that have the same result as PEI. So this is something that to explore on how PEI becomes the higher source of polymer in the beach sediment, whether they are deposited directly from the industry and ends up in the beach sediment. And this is the results for the green mussel. We have a total of 35 green mussels, and the plastic particles are found in 88.47% of the mussels. So the abundance is 0.45 plus minus 2.31 particles per gram, and in individual it's 2.77 plus 0.59 particles per individual. I compare it with other studies in Malaysia, Zahid et al. The concentration is slightly lower, but if you can see from the list, the results for these green mussels in Langkawi, there's kind of intermediate between within other states and other countries. And that is the results for the polymer characterization of microplastics found in green mussel. And it is very interesting, as we expected to see rayon as the higher source of polymer, as rayon was found to be the highest we found in surface waters. But in here we found PET, PP, and polystyrene as the highest of polymer. So probably another thing to explore is whether green mussels are retaining certain types of microplastics polymer apart from other types. And these are among the microplastics found in Langkawi. A is the fragment, comes in many different colors. B is foam, C is fibers, D is from pellets, and E is film. So for the conclusion, basically, microplastics are present in all samples, whether in sediment, in water and mussels. In water and mussels, fibers are the most dominant, while in sediment there are fragments. And the diversity of polymers actually stating that all the microplastic sources are coming from secondary sources. So most of it are actually coming from terrestrial environment, not from the marine environment. And it is a huge threat as these microplastics are already found in marine organisms, and what we have littered from the terrestrial ends up in our organism and we end up ingesting them. So it is vital for us in the future to provide extensive measures so that we are able to combat plastic pollution. These are, all right, that's all from me. Thank you. And I just wanted to express my gratitude to the organizers, to the NAM SNT Center, to JSS and Scope, because without the organizers, we won't be here with one objective, that is to combat plastic pollution. Thank you.
Thank you, Najiha. Any questions? So, how frequently do y'all eat those mussels? Is that used for consumption? Yeah. So, is it a very frequently eaten thing? Y'all, like how? Yeah. Is it like commonly eaten?
Yeah, it is considered one of the commercial bivalve species. So we frequently consume it in our daily lives.
Okay. So in the absence of any other questions, thank you very much. So just give you a certificate. So thank you very much to all the speakers. We close this session now.
Thank you, ma'am. Being the moderator, I would also like to thank our session chair, Professor Sharmila Jaisingh, for coordinating this session, and also the keynote speaker, Professor Praveen C.R. Ramurthy, who had all along come from Bangalore to present here, and Mr. Mohammad Reza from Indonesia with a very good talk on spatiotemporal variations, and also Ms. Ninaja Binti Mohammed from Malaysia explaining about distribution of microplastic. Now I request Dr. Satish Kumar from Department of Environmental Science to give a chairperson certificate. Okay. Thank you, ma'am, for chairing the session, a token of appreciation in the form of certificate. I once again request all the delegates and the participants to please give your resolution form with any changes or additions which you have added up to us so that we can implement it and conclude it in our concluding session with the resolution. And I also request all the poster presenters to please give your posters in the registration counter so that it can be displayed, and the posters are displayed. And I also request all the participants to have a look at the posters because it's a research work being done by research scholars and also MSc dissertation students as a part of their research. Thank you.
get a response from. You have not given me.
Oh, you did not.
I
I
don't.
give it. 1 second. It's with man has sent it and asked it. Now we require resolution. Resolution very good. It was arranged in 12:38. The resolution, skill one paper give the resolution. Most of them have not received this. They're asking now. ಯಾಕೋ ಕೊಟ್ಟಿಲ್ಲ ನೀವು ಬೇಗ ಯಾರು ಕೊಟ್ಟಿ conference Dear participants, the lunch has arrived. I request you to break for the lunch and reassemble once again at 2 o'clock for the technical session. Very good afternoon. I welcome you all for the technical session 6, and the session chair is Professor Ibrahim from Alexandria University in Egypt, and the moderator for this session will be Dr. Vadiraj Keti, Assistant Professor from JC Seher, India. Now I request Vadiraj sir to introduce the session chair.
Good afternoon, everyone. I feel an immense pleasure to introduce Professor Ibrahim Hassan, who is a professor in air pollution at Alexandria University, Egypt. He is a fellow of Royal Society, UK, and president of Scope, Egypt. And he is an air quality consultant at United Nations Development Program. He has more than 105 research papers and 4 published books, and he is journal for various environmental research publications. So thank you, sir. Thank you for coming and hearing this session. Thank you.
Good afternoon. As we heard yesterday, after lunch, everyone is sleeping. Everyone wants to go, especially this is the last session. But I'm happy because this session is addressing an important goal from the SDGs goals, Sustainable Developmental Goals, which is preventing plastics in environment, goal number 15. And I'm sure that we will have a good time during that session. We will start with Dr. Gauri Prasad from Colombo University, India. He will be talking about overview of plastic pollution on terrestrial ecosystem in India.
The presentation is for 10 minutes for presentation and 2 minutes for discussion. I request all the speakers to keep in time. Thank you.
So good afternoon, everyone. So myself, Guru Prasad from Colombo University. I am pursuing my PhD under the guidance of Dr. Basavarajappa SH. So today, I will be presenting my paper on overview of plastic pollution on a terrestrial ecosystem of Aganashini estuary of Uttar Kannada district of Karnataka, India. So coming to, as all the speakers have earlier told, so everyone know about material with almost limitless possibilities. So earlier we used for the long-term users, but now it is a part and parcel of human life. So plastics were mainly used for long-lasting items, but now plastic litter causes economic losses to the tourism, fishing, and shipping sectors globally. So coming to the point, estuary. Estuary is a place where river meets the sea. So in this case, I am choosing Aganashini estuary, which is to be announced as a Ramsar site within 2 to 3 months, and it is one of the last major free-flowing river of Uttar Kannada district. So the main objective of this study is to highlight the potential impacts of the plastic on the ecosystem services and climate change, emphasize the importance of plastic waste management, socio-economic impacts, and the role of community, and summarize the policy interventions to minimize the plastic pollution and plastic waste. So this is one of the review of literature where they have discussed the approaches and current recommendations for the control of plastic waste. So this is the place where we carried the work. So this is the photo taken during the low tide, Aganashini estuary, and this is the place where the river meets the sea. So methodology. So Aganashini is a one of the last major free-flowing river of Uttara Kannada district. It flows for almost 121 kilometers, starting from Shirsi and ultimately reaches the Aganashini village at Kumta town. So the periodic survey was conducted between September to December 2022, that is a post-monsoon period, where the fieldwork was taken in Aganashini estuary banks, which has an area of 3.5 kilometers. So random sampling was done at 10 different locations by using 10-meter standard net, and plastic collected in net was estimated in a weighing scale. So also we consulted local people and local environmentalists about the problems and possible solutions of this region and suggested some remedial measures. So this is our results and discussion. So during this survey, we found that average plastic deposition in the sampling site was found to be 1.8 kg. So for the area of 3.5 kilometer, the estimated plastic deposition could be 6.3 tons. So it was during the post-monsoon period, and it will vary for the every season. So our study revealed that the plastic flow from the source of water to end of estuary is affecting the estuarine ecosystem. So almost for 121 kilometers, the plastic contribution from the people at different places ultimately reaches the estuary, as it is a free-flowing river. So these plastics are also altering the water quality of the estuary, which indirectly affecting the livelihood. So my main focus was to discuss about the livelihood problems these people are facing. So around 300 families living in the Aganashini estuary are depending on the bivalve collection. So the microplastics affecting the deposition of calcium, magnesium, which affects the formation of shells, which needed for bivalve survival. So bivalves, so this is the image. So these people, mainly these people living in this Aganashini estuary are fishermen. So they are depending on the bivalve collection for their livelihood. According to 2008 study, around 22,000 tons of bivalves were collected, and 57 crore was the contribution to economy from the bivalves itself from the Aganashini estuary. But it was from 2008, but now the time has changed and everything has been decreased because bivalves are also not formed like earlier days. So as it is a Ramsar site, the large number of native birds, migratory birds, the livestock and soil fertility is also affected, as coconut and paddy are the major crops of this area. So there is a urge to control the flow of plastics. So we have suggested some, see, this is the images where this livestock cattle is feeding on the plastic. So we suggested supporting alternative to single-use plastic products, as there is availability of large number of bamboo in this region. So promoting reuse, creating awareness through the educational programs is also important. Regulating the uncontrolled tourism activity is one of the major threats for the Aganashini. So setting up the waste management unit in Aganashini village is also one of the recommendation. Strict government rules and laws. The distribution and concentration of micro and nanoplastics need regular monitoring. So this is an overview of the current state of science. So we recommend future research in this region to understand better understanding of the plastic debris transport in the estuary. So we need to conserve the last flowing river of Uttara Kannada, because the majority of the people are depending on the Aganashini for livelihood. So thank you. So this is the one of the child who is involved in collecting the bivalves. So this is what importance of the Aganashini estuary. Thank you.
Thanks for your punctuality. I appreciate that very much. I will open the floor for audience if there is any question. Any question? Well, so there is no question. Thank you very much indeed.
Thank you.
I'm happy to give you.
Thank you, sir. Thank you.
Right. So we will move to our next speaker, Miss Afika Kunain. She will be speaking about microbial degradation of plastics, a sustainable approach to tackle environmental threats.
presenter is not there.
All right. So we'll move to another, the third one in the row. Dr. Sumana, he will be talking about microbial metagenesis approach towards effective degradation of plastics from GSS Academy of Higher Education and Research. Dr. Sumana, yeah, she's ready.
Good afternoon, all. I thank all the organizers, SCOPE, NAM, SNT, and the Department of Environmental Science for giving me an opportunity to just share my views and thoughts and some of the areas of concern that would really help young researchers and young minds to just think about the aspect of overcoming whatever the pollution and the wastes that are going to be generated. So that is there any microbiological alternative so that can give you a very good insight to address these issues. So with that backdrop, I just selected the microbial metagenomic approaches towards the effective biodegradation of the plastics. So being a microbiologist, I'm not a material scientist or something like that. I just wanted to understand what are these plastics made up of, first of all. So when I was just surfing, it is starting with a story where a story speaks about the balls for billiards or the snooker, the game of bureaucrats, where the balls were made with a specific dimension, specific diameter, radius from that of the elephant tusks. Over the period of time, the availability of tusks and the preparation of these balls was not meeting to the requirement, and the company was the company which was producing those balls puts forth an advertisement telling that is there any scientist who will be able to replace the material from that of the ivory with that of any other material, so that who will be rewarded with a very huge amount of price. Then comes the celluloid, the component which was made of the synthetic material that was in 1890s, which was a material that was a component that was generated by using this nitrocellulose as well as camphor by John Wesley Hyatt in 1863, followed by this celluloid was not meeting the requirement, or it was not exactly like that of the ball which was of the ivory, where the bouncing and the structure, the movement of ball of the celluloid was not coping or equivalent to that of the ball that was of the ivory. And later on, it was also found inflammable as well. And then parallelly, or in the contrary to that, there was another research that was going on for finding out any other material which had this waterproof or leak-proof capability. The material which was, shellac is a component which is a resin type of thing, where the resin is the what end product where an insect comes, and such type of resin was seen to be happening or developing on the bark of the wood. So this shellac was used as one of the coating agent for many of the food material, so that the shelf life of the food material was increased because of the capability where the oxidation and reduction process in the food material or any other liquid that was supposed to be blocked. The leak proof or the oxidation reduction reaction or gas leakage was totally overcome by using this shellac. So this shellac in many of the food industries was not meeting the requirement. So parallel research was taken up to overcome the material so that could substitute the shellac. Then started the Alexander Parkus in 1862 he starts using synthesizing a kind of plastic which contained, which was a substitute for these shellac material. And later on, it was the bakelite. It was in 1907, a first synthesized plastic which was made out of this phenol and formaldehyde. And finally, it just takes into a shape while using many such materials like silk, wool, cellulose, and latex, etc. So why I just gave you all a brief backdrop of the history is because whatever plastic that is generated, that is synthesized, that is man-made, is from the natural substances that are available. I mean to say, the silk, wool, cellulose, latex, or whatever material, phenol, formaldehyde, all of them are naturally available, and they are usually, made to react, and a component is synthesized. Okay, so that means to say, basically, they are available in the the raw materials for the preparation of this plastics are just nothing but the things that are available naturally. Then comes after bakelite, it was found to be less inflammable, and later on in 1920, it was polystyrene and the vinyl, acrylic, nylon, polyethylene, finally, right? So when all these things are into place, so finally, the entire plastic substances was exploited, not only one specific field, instead in all the other fields. Now we will be able to say, like in microbiology, we say microorganisms are omnipresent, and now plastics are found to be omnipresent. So when we say omnipresent, obviously it has totally entered into the entire lifestyle of the human being, and it has totally, the demand for that has totally increased. And we have reached in such a stage where the plastics have reached to its saturation. Now, managing that is a challenge. Okay. So like in 2050, these are all the estimates, I think, since yesterday, you all have been heard of, you all have been, aware of. And now the plastics have been accumulated in such a way that it has reached to an issue which is of global concern. So this is what I think we have just spoke about since yesterday. And one thing is, it is just nothing but 40% now the entire globe is thinking of recycling. Now I'm concerned about the 60% which is not processed, which needs to be taken care of in future. So these are all different types of plastics, something like polyamylene, and these are all different types of plastics which have been produced, as well as based upon the utility. And this is all, the ones which have entered into the entire ecosystem, whether it is in marine habitat, terrestrial habitat, even in air, as we all came across, the plastics are, have volatilized, and they have entered into the nanoparticle size, and they are found to be present even in the atmosphere as well. So now these plastics, as I told you, are just nothing but the substances which are naturally available, and it is man-made. And how these things have to be degraded, okay, from microbiological perspective. So nobody till date, I think 1,000 years of research on this plastics is yet to happen. But at least we will be able to think of these things, something like recycling, incineration, all RRRs that we have come across till date. Like we can just think of the 60% unprocessed. So is there any kind of chances of processing and making it available so that it is bacteria friendly, and bacteria will be able to recognize this one as its food, so that it can be degraded or consumed, eaten, chewed by the bacteria or any type of microorganisms. So that will be converted into a simple form, and finally it will be utilized by the microorganisms for its energy and stuff like that. So that these plastics will serve as the food material, so that that can be degraded very well. So this is all about recycling and landfilling. These are all some of the approaches. Like it should not be mentioned something like China, Indonesia, Philippines, something like that. These are the, there are many such countries which are dumping the plastics into the oceans as well as rivers without any such processing. So what are the technological innovations? So Technological innovations, I think this morning we just came across the sensors that are usually developed to find out what type of component, what type of metal, what type of plastic that is found to be present in any other habitat. Likewise, from the microbiological perspective, is there any microbe that is available so that the microbes can be utilized, exploited? We can genetically engineer, we can convert the microorganism so that it can utilize that plastic as its food, so that it can be converted into the simpler form, so that can be managed very well. So this is one of the approach that can be taken up in future. So microorganisms can serve as a potential garbage cleaner. And there are so many kind of approaches which have been taken up by the microorganisms, which have been studied very well. When the microbe is capable of degrading the nails, the hair, and the way by which it degrades may take much time, but it is found to be promising that most of these microorganisms do have the enzyme systems, enzyme machinery, so that they can utilize the food and they will be able to degrade any complex molecule. So this when we study about these microorganisms, which are associated with the plastic degradation, are said to be the plastisphere. So we have to dig out the plastisphere and we have to analyze the type of microorganisms, because it is not one microbe which is involved in the decomposition. It is a consortium of microorganisms. And in the consortium of microorganisms, you have different types of microorganisms which are found to be present. It may be bacteria, it may be fungi, it may be actinomycete. And there are so many types of consortial bacterial species which are associated for the decomposition of the plastics. So we do have to find out, we have to find out what are the type of microorganisms. We should decipher the type of microorganisms and the efficiency of them. That needs the mechanism that is involved, the type of enzyme systems that is involved for the decomposition is to be focused upon. So this is just nothing but the pathways. Usually most of these consortium form a biofilm, which is just nothing but a polysaccharide slimy material, which makes the plastics physically as well as chemically weak, so that most of the microorganisms will be able to degrade this complex form of plastics into the simpler form, whether it is in the fibrillar structure and finally into the monomers. So that can be exploited for the construction or usage of so many other recycling kind of thing. So this is just nothing but what do you call the flow chart, where we will be able to attract and we will be able to find out the type of microorganisms which can be involved in the decomposition by the formation of a biofilm. So finally, after the complete decomposition, this is just nothing but the study by Glacier in 2019. I'm just sharing the kind of research that was done. Once the plastic are totally decomposed, these are all the end products that we'll be able to get, something like benzoic acid, benzyl alcohol, and stuff like that. And these oligomers and the monomers of plastics usually are assimilated. Assimilated means they are utilized by the microorganisms for the purpose of energy, for their growth, and later on they are catalyzed and for the sake of energy. So the assimilation helps in the conversion of monomeric plastic into various secondary metabolites, which later on are excreted into the environment, which will be environment friendly. So that means to say all these plastics are processed, are catabolized, are metabolized, and finally it is excreted out so that it is environmental friendly as well. So when we study all the catabolism or metabolism, directly it leads to the genes which is responsible. I think the audience are interested to listen to me. So I request the organizers to please give me some 5 more minutes. I'll just speak about the metagenomics. I hope the audience are okay with me. So the study was all about on the metagenomic studies. That means which gene is involved for the production of the specific type of enzyme systems. So those enzyme systems will help the researchers so that that specific gene is enhanced, the effectivity or the way by which the organism, the speed with which the plastic are totally decomposed, degraded and catabolized was researched upon. And later on comes the analysis, how actually
the genome analysis are usually being done, and the gene that is responsible for the production of specific type of enzyme systems, so that these enzyme systems will break down the bond in the polymer, and later on it will be converted into the monomer. So there are two types of structure, metagenomic approach. One is the structural as well as the functional. From the structural kind of perspective, it's just nothing but tracing the specific gene through the PCR analysis, molecular analysis, and through that of the functional approach, the specific type of enzyme pattern the microorganism holds, so that can be exploited, and the organism can be genetically engineered and can be made more effective, so that sometimes like the advanced studies has towards this metagenomic analysis is the system biology, where we will be able to synthesize the specific compound. We will be having the genome editing. We will be synthesizing microorganisms. We don't know. The research is heading in such a way that we may generate, synthesize a superbug. Generate a bacteria with the genome editing as well as metagenomic analysis, so that bacteria may be involved in the catabolism as well as the metabolism of this, the plastics. Okay, this is just nothing but the list. The ones who are interested, I can share my slide. These are all the list of microorganisms which are known, which have been published. Okay, are involved in the catabolism of the specific type of plastics towards the left. And these are some of the approaches which has been done. One is to manipulate the genome of like the genetic engineering, adopting the genetic engineering methodologies. We will be able to identify the organism, and we will find out what is the time that is required, and the microorganism is isolated. It is genetically engineered, its efficiency is increased. Later on, we are re-inoculating back, so that the time and the speed is totally increased and efficiency is increased. That is one approach. And the second, microbial genetic constitution, like it is a kind of design and a programmable set of thing, so that the same thing is applied to that of the environment, so that you'll be able to degrade, or you'll be able to achieve the thing faster. So these are some of the challenges. Like we will be going through the gene editing, as I told you, synthetic and systemic biology, using those CRISPR-Cas9 and TALEN and Zen. Okay, these are all some of the molecular approaches, and through that of the novel enzymes, through microbial metabolism and catabolism, so that we'll be able to generate the secondary metabolites, so that those secondary metabolites will be able to degrade the plastics. And there are certain, synthetic regulatory, the specific gene is identified, and through the genome editing, we can make the organism more efficient. And some of the, cell-free technologies and protein expressions, these are some of the studies that have been taken up now, again, to the degradation of these plastics. And the metagenomic technologies, which enable the development of proteins with the enzymatic activity, so that the degradation is enhanced well. So I would like to conclude, any judicious use will usually, the risk gets reduced. With this note, I conclude my session. I'm open for any questions.
Thank you, Dr. Sumana.
Thank you, sir.
I will open the floor. Any question? Okay, thank you.
Sir, ma'am, so what you are saying, genome editing is nothing but genetic modification, right? Yes. So how you are sure, how much you are sure that it's safe for environment? It will open, it's like a Pandora's box. If you open it, it causes some other trouble. So how can you justify with what, so solving one problem and taking up another problem?
No, very simple answer is microorganisms are very handleable. And unless and until you provide the food and you make that food, it can be consumed, it can be catabolized, you will, bacteria won't touch. That's the problem now since 2 days that we are talking about, because bacteria and microorganisms are not eating the plastics. It is, getting accumulated in the habitat. Okay. First, as well as the foremost thing is, before coming to genome editing,
It's the proteomics studies that we are supposed to carry out, and the way by which the biofilm that is going to be formed. And it is not only one bacteria, instead it will be a consortium of bacteria, and the type of enzyme systems and how the plastics are physically and chemically weakened is through that of the enzymatic studies is important. Okay, once you do that, you will segregate, you will just find out what type of organism is involved in the production of that specific type of enzyme system. And you will be dating back to the genome. And again, you'll study what is the time that is required by the bacteria to decompose that specific plastic.
Yes, ma'am. I can understand with all those things. But how sure you are that a genetically modified organism is safer for environment? So all you are saying is you are giving the positive side of it, but simultaneously we are seeing some superbugs and all those things in the environment. So you are saying that we have a solution, but simultaneously the solution itself is a problem.
It cannot be. That is what my answer is. Unless and until you provide food and you make the organism to eat it, I don't think so there'll be any kind of thing. So unless and until there is catabolism and metabolism, they can't overgrow, or they cannot magnify, or they cannot, cause any problems. So that is my answer.
Thank you.
Any other question? Okay.
One more question. It was in your paper, one of the paper which you said done in 2019, that when plastic is degraded, the chemicals which are being benzoic acid, something like. What was the microorganism used in that paper?
It was just a consortium of bacteria. It is not that one organism is producing these many end products. So I'll not be able to say, or even in the paper it is mentioned that it is this E. coli has produced these end products. No.
So it was just
a
consortium.
consortium, and the biofilm which was formed is a biofilm formed by many bacteria. It may be fungi as well. Some of the actinomycetes, okay, may also be present.
Was the consortium cultured or collected?
No, it was just a study. I have not carried out any research. I'm very sorry for that. It's in the paper. It says it's the bacterial consortium which had the biofilm.
Thank you.
So even there, it's not mentioned that this specific bacteria was involved.
Thank you, Dr. Sana.
Thank you so much.
Thank you very much indeed. Right. So Miss Afika Kunain has arrived, so we will go back to her. She will giving us a talk about microbial degradation of plastic, a sustainable approach to tackle environmental threat.
A very good afternoon to each and everyone gathered here. I am Afra Kaunain from first year BSc Biotechnology, and our topic for the day is microbial degradation of plastic. Before we dive into the topic, let's know what plastic is. Plastic is a polymeric material. Polymer refers to which has repeating units of monomers. In the case of plastic, it can either be found naturally or made synthetically. A good example for synthetic plastic would be polyethylene. Plastic can be classified into two groups, LDPE and HDPE. LDPE is low density polyethylene and HDPE is high density polyethylene. As the name suggests, low density. Good examples for LDPE could be plastic bags or wrappers, which we use in our day-to-day lives. Examples for HDPE could be plastic pipes, chairs or jugs, etc. The production of plastic has increased exponentially in the past 2 decades due to the changing human lifestyle and needs. This has resulted in plastic pollution or improper disposal, which has negative impacts on the system. Biological estimations have shown that millions of aquatic and terrestrial organisms have simply died by ingestion of plastic, which was disposed improperly. We all can see in the graph, like in the last 2 decades, there is exponential increase in the plastic pollution. For the Few microorganisms have found to have degrading properties for plastic. Me and my team, along with the guidance of our professors, have done a research on fungal and bacterial degradation. The following mentioned strains have been used. Plastic pollution is causing a severe environmental issue because improper disposal in water resources has led to disruption of aquatic life cycles, whereas the plastic debris which was deposited in sewage systems has blocked the drainage, and it is breeding grounds for mosquitoes and other disease causing pathogens. The floating areas which can persist for years and years if the plastic hasn't been taken out. Plastic pollution is such a big issue because it is non-biodegradable, hazardous, and toxic if not disposed properly. Burning plastic is one of the solution, but it also has negative impact. It can lead to the release of pollutants such as furan, which are very harmful and toxic to animals and human beings. Plastic has chemically toxicated human and animal genome. It can cause diseases like genetic disorders or cancer. Polyethylene, as mentioned before, a synthetic plastic, is one of the most toxic form of plastic because it has a very slow degradation rate. The slow degradation rate is due to its water repellency and hydrophobicity. Not only is plastic non-biodegradable, the other causes for its non-biodegradability could be pH, moisture, the environmental soil, etc. So as I mentioned before, we have researched and worked on fungal and bacterial strains and looked at their degradation properties. The degradation of these fungal and bacterial strains was measured using like the difference between the weight of the plastic before and after degradation. These were further subjected to SEM and FTIR test, which also gave us the changes in the chemical structure and their morphology. So let us move on to the methodology or procedure in which the bacterial degradation has taken place. First, we had used 3 sets of pre-sterilized petri dishes in which we poured the agar medium for bacteria and PDA as well as agar for the fungal strains. These after solidifications were inoculated with fungal and bacterial strains respectively, after which the LDP sheets in the measurements of 6 into 6 centimeter were cut, sanitized thoroughly to avoid microbial contamination, weighed in digital balance to see the initial weight of each piece, and placed carefully in the petri dishes. They were inoculated for a period of 10, 20, and 30 days. These are the following fungal and bacterial strains which showed results. After the inoculation time, these were carefully taken out and weighed again, cleaning it with ethanol. The plastic strains which showed the difference in their weight after the degradation were subjected to SEM and FTIR analysis, which also confirmed the same. You all can see the results over here. The bacteria are mentioned at the right corner. In the left side shows the graph of 20 days degradation, and the right side in each graph shows the 30 days. As you all can see, in the 30 days of the first graph, F2, which is Escherichia coli, has showed a high amount of degradation in plastic. F and G here is annotated for food wrapper and gloves. These were the 2 forms of LDPE which were used. And these are the graphs for fungal degradation. These are few of the pictures of our research which were used for the fungal strains. Here are the FTIR results which we got. In the first one, you all can see that it refers to F control. F control and G control were 3 petri dishes which were kept aside. These 3 petri dishes were not subjected to any kind of microbial strains. This is because we wanted to see if nutrition alone on itself was capable of degrading plastic. And yes, we did find results. F control refers to food wrapper plastic, which showed minute degradation. The peak refers to alkane bonds. There is only one kind of bond present in the F control, which shows that there has not been any microbes, so there won't be any change. But in the second graph, you all can see it is mentioned over here. There are alkene bonds over there, and aliphatic amines are present. This is due to the microbial degradation, which has resulted in the change of the alkane bonds to alkene and aliphatic amines. The 10 days results, we didn't find much of the degradation. So most of the degradations were found in the 30-day analysis. With this, I can conclude my presentation by saying that we were successful in finding few strains of bacteria and fungus which could degrade plastic. Thank you.
Thank you. Any question? Okay. Okay.
The experiment, good presentation. The experiment was carried out for how many days?
Ma'am, as I mentioned before, it was carried on for 3 periods of time. One was for 10, 20, and 30 days. That was for bacteria. But in case of fungus, it was subjected to 20 days.
And the remove, I mean, how do you conclude? Conclude for 30 days was the complete degradation, or you achieved within 10 days?
No, ma'am, there wasn't complete 100% degradation, but there was slight changes in the weight of the LDPE sheets when we compared with the initial weight. So the LDPE sheets which showed difference in weight, those were subjected to SEM and FTRI analysis, which showed through electron microscopy that there was indeed a few minute pores which were not visible to the naked eyes, ma'am. Like there was degradation present over there.
Okay.
Thank you, ma'am.
You choose some of the particular strain.
Yes,
no?
yes, sir.
So, in what way you have chosen that?
Sir, some of these.
Is there any importance or any?
No, sir. Few of them, as you have observed, those were Salmonella typhi. We just wanted to say that these bacteria, as you know, can cause diseases. So we wanted to see if they have degrading properties too. Few were disease causing, and others were the ones which we obtained from our biotechnology life sciences department, sir, which were available over there. We obtained those.
So the good presentation. Then I wanted to ask some clarification. So because you use different microorganisms. So where did you get these microorganisms?
From our life sciences department, sir. Those were already present, like isolated over there. So I'm not sure about from where they have been isolated.
Okay, because these organisms sometimes can cause the diseases on plants as well.
Yes, yes, sir.
So did you check that properties of these?
No, sir. I'm sorry. We didn't check because it was already isolated in our department. We used it directly, sir.
Okay, thank you.
Okay. Thank you, sir.
One more question here.
May I know the degradation efficiency of bacteria and fungi?
Yes, ma'am. As you can see in this graph, ma'am.
Is it with respect to days you are telling?
Yes, yes, ma'am.
Yes.
The left picture figure shows the 20 days. For fungus, both were 20 days, ma'am. This left one is for the food plastic. And here we can see that F17 strain, which is Fusarium species, has shown the maximum results. Whereas in the right, G18, which is Penicillium sp, has shown the maximum results, ma'am. And in case of bacterial degradation,
can you combine like consortia like bacteria and fungi? You can combine and degrade. Is there any chances of that?
Maybe we can, ma'am. In our future research, we might try that with different strains of the ones which show degradation. Thank you, ma'am.
Nice one.
Yes.
What is that y-axis? In the previous slide also, there is no legend for x-axis. X-axis we can say, but what about y-axis?
Sir, y-axis is the amount of like efficiency of the degradation, sir. You can.
So how can you calculate efficiency? Exactly. How did you calculate the efficiency of
This one was subjected to SEM test, sir. So it is according to that scanning electron microscopy.
So what you are showing for that F2 for 30 days, that's already 100%.
It isn't percentage, sir. It is like the number of pores or difference in weight, sir.
So 100 grams difference, or
No,
what
not
exactly
grams.
is the difference? What you are showing there?
the electron microscopy, sir, like the amount of beams we received after subjecting it to SEM test, like the intensity of beam, the electron beam.
Thank you.
Okay, thank you.
Thank you.
Hi, hi, hi. Right, we are moving now to the last session of today and last session of the workshop. Miss Gaya Sairi from Fatima Mata National College, Kerala. She will be talking about microplastic pollution in the sediments of Ashmudu Lake, Kerala.
Good afternoon, everyone. Today, we came to the last session of the two-day international workshop. And for 2 days, we have dealt with different topics about plastic pollution, microplastics, their consequences, mitigation, and all. Now, I'm going to present a topic of my research work, microplastic pollution in the sediments of Ashtamudi Lake, a Ramsar site in Kollam district, Kerala, India. And I'm Gayatri, research scholar, Department of Zoology, Fatima Mata National College, University of Kerala. As we all know, plastic is present everywhere, and it's a essential part in daily activities. And it is estimated that around the year 2050, there will be about 12 billion tons of plastics in landfills and in the environment. So, due to the non-biodegradable property of plastics, there is a buildup or accumulation of plastics occurs as more and more is released into the environment. These long-lasting plastic trashes were mainly divided into 5 categories based on their size: megaplastics, macroplastic, mesoplastic, microplastic, and nanoplastics. So we are dealing with microplastics. Microplastics are tiny pieces of plastic which have a diameter within 1 micrometer to 5 millimeter. Based on the origin, they are mainly classified into 2 types. There are primary microplastics and secondary microplastics. Primary means they are originally manufactured in a particular size for specific application, such as in toothpaste, facial scrubs, etc. Secondary microplastics are formed as a result of breakdown of larger plastic debris by photochemical, mechanical, and biological process in the environment. So these microplastics can be seen in different shapes, such as fragment, pellets, fiber, foam, microbead, film, etc. And the most commonly found polymers were high density polyethylene, low density polyethylene, polyvinyl chloride, polystyrene, polypropylene, polyethylene tetraphthalate, etc. So coming to the sediments, they are the important part of aquatic ecosystem, which serves as a home for many different organisms and plays a role in nutrient cycling. However, these presence of microplastic in sediments have some negative effect on this ecosystem. These microplastics can accumulate in sediment over time, which leads to the potential for long-term exposure for organisms which live in these sediments. Study site is Ashtamudi Lake. It's the second largest wetland ecosystem of Kerala, and it is designated as a Ramsar site in November 2002. And it has an international importance for its hydrological functions, biodiversity, and faunal diversity. And due to population density and urban pressure, it poses threats to the site, including pollution from oil spills from fishing boats, industries, and the surrounding areas. And according to Kerala State Pollution Control Board, Ashtabudi Lake environment has been negatively harmed by the uncontrolled dumping of solid waste and sewage from homes and businesses. As these rivers and streams flow through densely populated urban stretches, they can be the possibility of occurrence of microplastic is considerably high. So we take this local population, as the local population largely depends on this lake for this livelihood, I selected this as the major study area of my project. So the main objective of my work is to verify the presence of microplastics in the sediments collected from various sites of Ashtamudi Lake and to identify the abundance, type, and composition of microplastic using FTIR spectroscopy. So coming to the materials and methods, we collected samples from 6 different sites of Ashtabudi Lake, 3 sites which were located in the urban zone and the remaining 3 from the rural zone. And these are the sites selected. And sample collection was done during post-monsoon season, and 3 sediment samples were collected using graph sampling method, and for sampling procedures were done according to AFA 2012, and the plastic bottles were completely avoided during the research, and the collected samples were transferred to glass bottles and transported to laboratory for further examination. And these samples were sieved through 5 millimeter mesh in order to remove the large debris and to retain particles of less than 5 millimeter in size. So the sieved samples were dried in glass trays, and the extraction of microplastic was done as per National Oceanic and Atmospheric Administration Protocol by Masura et al. 2015. So coming to the microplastic extraction and polymer identification, this dried sediments were wet peroxidized according to Lee et al. 2016 using hydrogen peroxide, and the digested samples were density separated by the method of Galgani et al. 2013. These floated particles were filtered using a Wortmann filter paper and is visually sorted using a stereo microscope. So in the stereo microscope, we can see several microplastics or of different colors and different sizes. And these particles were taken for polymer identification using FTIR spectroscopy. So the results are, all the sediment samples collected from the lake were detected to be contaminated with microplastic. The average abundance of plastics ranges from 104 plus or minus 1 to 918 plus or minus 1 items per kilogram, indicating the extensive dispersion in the lake. Among the stations, site 1 showed higher number of microplastics and 5 different morphotypes of plastics were found. Mainly, fiber is the most common one. 33.37% of fiber is identified, followed by fragment, film, pellets, and foam. And analysis of statistical difference in microplastic abundance between 2 groups was done using one-tail t-test, and the results revealed that the urban areas had significantly higher abundance of microplastics than the rural areas. So these are the table containing microplastic abundance and polymer types obtained. Microplastics of 6 different colors are obtained: red, black, green, yellow, blue, and colorless. And the polymer components that were identified were polypropylene, polyethylene, polystyrene. And polypropylene was the most abundant polymer. There is 46.63% of polypropylene was found. These are the graphs showing abundance of plastics in based on classification based on type and color. These are the FTIR spectra of polyethylene, polypropylene, and polystyrene. So these are the stereo images of different shades, fragments, pellet, etc. So coming to the conclusion, the present study provides an evidence for the first time of microplastic in Ashtamudi Lake, and the results are that they are ubiquitous and abundant in the sediments of Ashtamudi Lake. And the present study tried to identify the presence, abundance, distribution, and types of microplastics of Ashtamudi Lake of Kollam. And most of the microplastic were found in the site 1, which is Asramam. It is one of the prime centers of Kollam city, as it is one of the best tourist spot in Kollam. And there were so many parks, hospitals, industries were located in the banks of Ashtamudi Lake. So hence the pollution level is considerably high in this area. And compared to other sites in rural area, abundance of microplastic is considerably high in a site called Samranikodi, as it is an emerging tourist sport, a lot of human intervention is occurring. So four different morphotypes were obtained, different colors were obtained. This presence of polymers like polypropylene, polyethylene, etc. Indicates the plastic pollution on and off land due to plastic carry bags, plastic bottles, packing materials, which needs an urgent management action. Ashtamudi Lake is a source of livelihood for thousands of fishermen and is stated to be the second biggest fish landing center after Vemanad estuary in Kerala. So it's important to make environmentally relevant approach to understand the fate, behavior, and impact of microplastic as an environmental pollutant and decrease the risk of pollution. So this study is an eye-opener on microplastic pollution of Ashtamudi Lake. We express our sincere gratitude and thanks to University of Kerala for providing funds for the research work, and to the director and technical staff of CLIF for the guidance and assistance in the HDR FTR analysis. And I also thank the organizers of this event for giving us the opportunity to present our work. Thank you.
Thank you. Any question?
Yes.
Okay.
In which site did you find highest amount of microplastics?
Site 1.
Site. Yes. Okay, fine. Did you check any effects of these microplastics on aquatic animals in
Yes,
the
I am working on fish and also bivalves in mussels and oysters also.
Okay. On a brief note, what and all the problems which are being faced by those organisms? Can you?
So now I am doing the preliminary study of how the abundance of microplastic. The biology study is I am doing it in the next test.
Okay. Thank you.
Okay, another question.
I have one question. So you have told about in sediments, the microplastic accumulation. So we can able to see only specific type, or if you can able to see many or multiple categories of microplastics? Only specific type you could able to see, or any other classifications or specific classification you have observed?
Now we have obtained 3 polymers of microplastic.
So out of those, which one is the most dominant one?
The polypropylene is the most prominent.
Polypropylene. Okay, thank you.
Any more question? Okay. Thank you.
That was the last presentation of this session. I thank all the speakers, Dr. Sumana, Mr. Guru Prasad, Dr. Arfa Khanun, and Ms. Gayatri for her presentation. I thank Professor Ibrahim sir for chairing the session. I request Pallavi madam to provide give the certificate and memento. Now we will arrange for the panel discussion, and panel discussion will, the members of the panel discussion is Dr. Bandyopadhyay, Dr. John Senset, Dr. Nivels, Dr. Ibrahim Hassan, Professor Manoj Kumar B, and this panel will be coordinated by Dr. Shivraju HP from JSS Ahir and Dr. Suryanarayan S from Scope.
Thank
Hello
you.
from Cape Town. Can you hear me?
Oh,
panel discussion and panel discussion followed by adoption of resolution. So for this panel discussion, I request Professor John Samset, President Scope, is going to chair this panel. Along with that, Dr. Amitav Bandyopadhyaya, Director General, NAM SNT Center. Dr. Navile, Vice President Scope, is going to join this panel discussion through online. I also request Professor Ibrahim Hassan, Professor from Egypt, to join this panel discussion. Also, I request Kandaya Fakirnathan, University of Jaffna, Sri Lanka, to join the panel discussion. And I request Dr. Sarvamangala Pravina, University Putra, Malaysia, and Professor Sharmila Jayasena, University of Colombo, Sri Lanka, with the member of this panel discussion. So I request Professor John Samset to chair the session and initiate the panel discussion. Over to you, John.
Hello, I'm looking over your shoulder.
So, we shall start the panel discussion. So I request Professor John Samset to initiate the panel discussion.
Thank you. It's good to see that there are so many people here at the end of the conference. Usually it's not. Yeah, so it's really good. And then I hope we'll have a fruitful panel discussion. And I guess the members will introduce them. So what I like to do is that we'll start with an opening statement from each of the panel members. So why don't we then start with on my right side.
Good afternoon. And as John has said that, okay, at the end of the day, second day, the house is nearly full. That is really good. Excellent. It has a bigger clap. Okay. As we have listened to many lectures, one thing is clear that the problem is much more serious than people anticipate. Whether it is handling the plastic pollution logistically or getting into a deeper look about the academic aspects, that what is exactly happening, including the mechanistic issues. Those are the other angles. My career was in research, and by training, I am a chemical engineer. And I have worked on the area of environmental pollution for about 15 odd years. But those are the environmental pollution with the metallurgical industry. It is a material, but no, I've never worked on the plastic, anything related to plastic. See, what I would feel is that, and I was going through the posters yesterday and today, and so much of good work is being done in JSS. And yesterday, you also must have seen that our Honorable Chairman of the Karnataka Pollution Control Board, he was really very happy that, okay, such an issue we have taken up, and this should be taken forward. What I excellence on plastic research. I would rather say not plastic research essentially, research on plastic pollution. Initially, the center can start as something like a virtual activity, as a virtual center, because setting up a center requires money. So to start with, and creating a website, a platform for sharing information doesn't really need a lot of money. All those expertise are available. So what my humble suggestion to this forum, to all the experts who are sitting here, is that JSS Academy of Higher Education and Research can initiate this and bring together all the research activities that is going on in JSS, as well as other institutions which have participated. Some of them have presented here, but there are many other institutions which are not present today. So first create a database that all these institutions which are interested in joining the particular center, and then create a virtual center first, and then subsequently talk to the JSS authorities and other decision makers, so that the Center of Excellence can be set up here over the next 2 to 3 years period. And from the NAM Center side, I can say that, okay, you can include the name of the NAM Center as a part of the Center of Excellence. And I would also request John that if he also agrees that scope can be a part of it. So that would be a wonderful initiative. And it will go a long way in bringing the clarity of the plastic pollution problem that we are currently facing. Thank you.
Good afternoon. I was so happy to see young researchers are much interested in the topic, and I've seen many posters, and they are very promising, and they have the potential actually. We heard about 25 lectures. One of them was favoring plastic matter, and the 24 was anti-plastic. So we respect both of points of view. Both are correct. The guy who said that plastic is good and plan, not ban, it's good idea. We have to respect, we have to understand his point of view very well. But we are suffering from plastic pollution. We are the people who are creating the problem. So we have to think carefully how we can solve this problem. I think from discussions over the last 2 days, yesterday and today, we reached to a reasonable answers and reasonable research methodologies to tackle this problem, either biologically or chemically or physically. So I'm sure that this workshop is one of the best workshops I've ever attended in my life. It is very promising. The ideas are verified and different aspects, and they discussed many problems, and they addressed also not only the problems, they addressed the answers. So I wish you all the best, and I hope success for everyone. Thank you.
No. So now I want you to talk behind my back, Neville. It's your turn.
Thank you. Can you hear me?
Yes. Yes.
Good. So I'm not very prepared for the panel discussion, and but I do always have an opinion, and I'm going to be a little philosophical, if I may. And I want to say that the plastic pollution problem is not a special problem. It's just one aspect of a very complex set of problems, all of them driven by one or two common drivers. So I believe that what we are suffering from in plastic pollution is a consequence of overconsumption. It's a consequence of capitalism and materialism. And I think these issues have driven us to overconsume and to look for convenient ways to do that, which result in several environmental problems, so serious that they are, in fact, existentially threatening our future. So plastic pollution indeed needs special attention. It needs to be focused on to find solutions to the problem. But I believe the solutions are multifaceted. The solutions lie both in a systemic approach to environmentalism as well as the practical approach applied to plastic pollution. So I was thinking earlier today, 100 years ago, my grandparents, they never used plastic. They used brown paper bags, recyclable or at least organic bags that can be managed quite easily to carry their groceries. They used glass bottles or earthenware bottles to carry liquids. And now we've become addicted to the petrochemicals, and not just for plastic, of course, but for everything else in life. And of course, we need that. We need progress, and we need to find a way to continue to grow and develop humanity, but we have to take cognizance of the price of that. So to me, the solutions are indeed, research is necessary. We can't do this without knowing what the problem is, how the problem is trending, what are the major impacts, and how to manage them. We absolutely require research. We also require good policy and regulation, because without that, we can't control what people do. But both of those are not solutions to the problems. Policy and regulation also requires enforcement, and it requires monitoring. And to expect to solve the problem with research and policy, I don't believe is going to take us to where we need to be. The changes that we need are social changes. We need to ensure that people are properly educated, and once they are educated, that they have the means to make decisions in a democratic way that see the results that improve their lives. It can't come from the top down. It has to come from the bottom up. We need to address the root causes of poverty, and we need to address the root causes of excess and of overconsumption. And people who are educated will then make powerful and important personal decisions that will see this problem resolved. So that's my statement. Thank you.
Thank you very much for giving me an opportunity to serve as a panel member of the panel discussion. For the last 2 days, we were enjoying and the different research findings presented by the different experts and the scientific groups, from the origin of the plastics until to how you can manage the plastics. So different groups are working to manage the plastic. For example, the chemistry people, they are working into produce an oil and in different ways. And the biological scientists are identifying the different microorganisms to decompose these microplastics. These all are mainly in the experimental level. So in my point of view, because from this many presentation, it says we cannot omit the plastics because it's giving day-to-day, it's attached to the human life, the day-to-day activities, as well as it's giving an economy for the poor people. So therefore, in a way, if the scientists are working towards to move this research finding from the laboratory level to the field, I hope it would be would give a good solutions to combating these plastic pollutions. So this is my kind request to the research scientists who are working here to align that research into chemical degradation and the biological degradation. Then, so as usually we are doing for the composting process is the mass scale, like that if you collected plastic waste, whether we can do for the mass scale composting process through the chemical process or the biological process, it is not until now it's lack. So in such a way, if you find the research alignments, then we can achieve our aim and objective of the workshops. I hope that's it. Thank you very much.
Thank you. So this is my first interaction with NASTIC, and I thought I was a fish out of water when I came here initially because I'm not really in the environmental pollution field. My field is microbiology, but I realized that that is very much part of the problem or solution to it. So I see it's a really good forum where you get exposed to all the different aspects. And I think it's very good exposure for the research students as well. And I saw some really good papers out there. And I realized that all the different aspects need to be considered. And there is the removal that is a field I am in. So the biological removal, as well as I've seen a poster on the bioplastics as a replacement. So we need to have the alternatives. The alternatives are very important because nothing's going to happen unless we have an alternative. So I think that is one area that we need to really look into. Thank you.
Good evening, everyone. So being in this international workshop actually opens more research ideas to me because from all the research finding that all of us shared each another, looks like it's actually a global problem, not only in a certain region, but it's all over the world. So besides understanding the problem, this workshop has also opened research of solution. As I see, sensors are one of the promising way in minimizing time to analyze microplastics and also other techniques available. But besides all these research findings and research ideas, I think importantly is to be able to deliver our findings to public. I think I found it very challenging to tune the research finding, scientific research finding, bring it over to public. I think that's another a way that we need to think so that this workshop output can be delivered to public so that they will also be aware the impact of this pollution to terrestrial environment.
Thank you. So are there any comments to the what the statements here? Are there any comments to any of the statements?
Okay, now based on the comments. Yeah, based on the comments made by other panel members, if any of the other members would like to add something to it, I think that we can consider. And then we'll summarize all these points, which will come out as a report. Like there would be a complete report on the whole workshop, in which we will also have a separate section dealing with the report of the panel discussion. That what the panel members have uniformly agreed on certain recommendations. And then after that, we will go for the resolution session. That is the idea, John.
Okay, now I have a couple of questions for the panel. And that is, in Norway, I have a colleague who has been working very deeply with plastics. And some 10 years ago, he told me that, it's really strange, because until now we have been spending all our research effort into making better and better plastics. But now we have to spend our research effort into making poorer and poorer plastics. So the question is really the idea of a biodegradable plastic a good one? Because we know that it is not truly biodegradable, and it creates a lot of microplastics compared to the old-fashioned plastics. So I'd like to have your comments on that one.
You can take. I have no expertise on that.
Right. As Professor John said, biodegradable plastic is not a real thing because it gives rise to micro and nanoplastics which dissolves in the soil and go through the food chain, and it gives hazards for the health of ecosystem, health of human beings. And I've read a paper saying that there are nanoplastics in the blood of people when they are making some kinds of medical checkup, normal analysis. They found lots of nanoplastics. So I think the traditional plastic, although it's very big and it has a huge problem, but much, much better than biodegradable because we can see it, it's there. But the microplastics or biodegradable plastics, it has been degraded into small fragments which can go through the air, and we believe that there is air microplastics now, and we can see it in the air, not in see it, we can detect it in air. It has been loaded over particulate matter, so we call it MPPM, microplastics over particulate matter, which is very dangerous because, as we hear, it is carcinogenic, has fluorines, lots carried on particulate matter. So I think we have to think carefully about something else, biodegradation of microplastics, and if we suffer, if we force it to use biodegradable, we have to think carefully how to get rid of these small fragments, nano and microparticles.
Are comments from this side?
For example, if you are looking into the bioplastics, truly bioplastics until the experimental stage, in our university, the biochemistry department, they invented the bioplastics, and now it has been commercialized to the company. So now they have started to producing these bioplastics as an alternate source for these synthetic plastics. But in success rate and the cost, if you are considering that, that is, we have to spend too much. So because the cost for the producing of the biodegradable plastic is comparably higher than the synthetic plastics. That's what it may not be affordable for the consumers is the low prices. But still, the research findings are there, but the production of the cost is high. That's my point of view. I have seen that.
I wonder whether the cost will come down with time, or is it the research that is costly now, or is there a possibility that in time the cost will come down? That's what we have to look at. Right?
Neville, do you have any comments on the?
Thank you. Very interesting discussion. I mean, again, we have to do something, and I think the research community indeed needs to find alternatives. The question about the costs is a really interesting one. I mentioned in the talk I gave this morning that in South Africa we passed a regulation to give a minimum thickness to plastic bags in order to make them reusable. And the government seeks to increase that thickness, but there's strong resistance from labor unions as well as from the plastic producers precisely because of the cost. To make thicker bags means they have to re-equip their bag factories, and as a consequence of that, they would lose jobs and it would be a big capital investment for industry. So they're resistant to it and have won the argument, in fact. But I guess at some juncture, we have to make a decision about whether the increased cost is more than the cost of impact. And those are sacrifices and difficult choices we have to make if we are going to make progress in this problem and other problems. And certainly, again, I'm going to use South Africa as an example, as the one I'm familiar with. Consumer pressure has driven change. So it is very difficult in Cape Town to find somewhere where you can buy a drink with a plastic straw, because nobody wants to drink a drink with a plastic straw. They want a paper straw, or they want a metal straw, or they want an alternative straw. And I think these are the kinds of things we can do. I think it's very important that the researchers come up with alternatives, viable alternatives, albeit they more expensive, but that we make those available so that consumers can make the choice. And then we will see the changes we can get through that mechanism. Thank you.
Thank you. So then I have another question for you, and that is in Sweden, there is one factory that makes every 8 beverage carton in the world. Every 8. So it's an enormous machine. It's 400 meters long, and I've been there. You almost have a car to operate it, and that is, they're using the product out of that machine as replacement for plastics. The problem is that it has a plastic coating inside the paper in order to make it waterproof. So, and then the second problem comes, it's impossible to recover it. So it has to be burned. So is it better then to go for this alternative, which creates new problems, or stick with the plastics for bottles, which we can try at least to recover and reuse? Any comments?
Yeah, it's actually what he said is true, but there is an alternative we can think of, because as our research students and research scientists are here. So normally the honeybee, if you look at the honeybee hive, so the honeybee hive, they are making the homes. It is an waterproof, so water cannot go in. So the bee waxes are a lot is secreted as a natural secretions. If the biodegradable cellulose materials are lot, if we, the scientists, chemical scientists, can make to mix it with the bee waxes and make it as a material for alternate for the plastic, it would be a nice idea. So we can easily and cheaply we can produce plastics.
Jan?
Looking to natural environment for answers is a good, the mimicry is probably where we might find some answers. Biomimicry.
John, yeah, in solution to your problem, I think other delegates have already spoken about. Just another example, we have a very famous a marathon run here, and they use little plastic sachets of liquid to rehydrate the runners. And this year, they produced those packages of liquid, not with plastic, but with a derivative of a polysaccharide derived from seaweed. And so they're using alternative bioplastics, they call them. They're not plastic. They're made from a polysaccharide a natural polysaccharide. And I think there are two ways to solve this problem. One is to, well, maybe three ways. One is to use bioplastics as opposed to petrochemical plastics. The second is to use other alternatives. So, for example, you and I buy your company, your Swedish company's milk in similar cartons, which are cardboard lined with plastic. Now, I have two options there. One is to not buy them. So I don't buy those cartons. I buy plastic milk in plastic cartons, which can be recycled. But the third option is to use glass. And when I grew up, and I'm sure for many of us in the room, unfortunately, when we were children, milk came in glass bottles, and those glass bottles had redeemable tax on them. And why don't we go back to that? Why don't we use glass bottles with redeemable tax? It is more expensive to process. There is more labor involved, but it's worth it because of the benefits to managing plastic pollution. Thank you.
Thank you, Neville. Now I think it's time for opening up for the audience. So we like to invite questions, comments from everybody.
Thank you, John. I am concerned. Actually, the question may be answered by Professor Ibrahim. So, as we know, yesterday there is one talk, very good talk by Vasudevan. He emphasized about like not to ban plastic, but you can plan. So, if we find some alternative for plastic, maybe you are talking about biodegradable plastic, material and all. If you see the cost of that alternative material, you know, it may not be. Some concept topic.
Right. I think the best way is to tackle the problem of plastics. And there are many ways to tackle this problem. One of them is using pyrolyzers. So we have to change the waste into energy. So there is no waste actually. It is costly. It is just in a preliminary stage. But there are some countries like Saudi Arabia, like China, like Japan. They started to incinerate internally the plastics, all types of plastics, to generate oil, which is used as oil for motors, not for edible purposes. So I think the best way is to think how to use these wastes instead of throwing them or dumping them or recycling them. Even if we are, like Neville said, we recycled the bottles many times and then we have to throw it away. Instead of throwing them away, we have to use them as a source for energy in pyrolyzer, which is a good idea actually.
See, this is an excellent idea about incineration, but so far designing those incinerators are concerned, we have to be very careful because during the incineration process, a lot of chemicals are produced which are carcinogenic in nature. One solution which the Russians have worked out, I think Russians, the Moscow Institute of Steel and Alloys, I think they are the one. So they wanted to incinerate it and then use this gas, whatever is generated, as a part of a iron making process. But again, economics is a major factor. So even though they tried their best, and technically there was a solution, but economically it didn't see the light of the day. Yeah.
So if you look at the plastic, it's because the plastics we are getting a product from the petroleum. The petroleum, how it's originated. So this is because of these deep weathering and degradation process of all the living organism for a long time. So I hope that this, if you allow the microorganisms as a degrading as a process, it could be a good solutions for degrading the plastic materials in a sustainable way without producing any chemical substances. That's how these people are working for the biofilms and the microorganism for superbug and development. This could be an, I hope that efficient and alternative way to degrade this plastics waste, I hope. This is my mind, yeah.
Fine. If you want to ban the plastic, are we ready to ban the plastic, and are there alternative materials are available to use as an alternative for plastic?
It's obvious it's costly. It's obvious it is costly. Alternative materials, and then we have to go for again the cotton material or things. So it is can't be a perfect alternate for the plastics. But if you are finding a way to how we can quickly degrade the plastics, then we can produce the plastics. That is the way we have to think in a way. Yeah, I hope. This is my.
Okay. Thank you.
Are there already comments or questions?
Doesn't seem to be. If you don't mind, I have another question. It is regarding, of course, ultimately, the plastic management is individual behavior and attitude, isn't it? See, if I'm not correct, even Masudaran was telling one example, I will keep some chocolate and just I will observe how they are going to discard that plastic waste. Have any standard, tool or something, to change the individual behavior? Now, if we change the individual behavior for sustainable or proper disposal of plastic, even we need not to think about the ban of plastic. I just need some comment from, panel member.
I hope that the sustainable way of plastic management, if you are not allowing to leak the plastic into the environment, perfect collection could be the good answer for plastic management.
Yes, sir, I agree. Ultimately, the individual behavior matters here. I will use, if I manage, if I discard the plastic waste into the dustbin or a proper place where I supposed to be discard, fine. But I use plastic, I discard wherever I use. That is the issue here. Ultimately, it connected to individual behavior.
It's actually more than individual behavior also, because at the end of the day, the collection has to happen at a government level or municipality level. I mean, in my own experience, I collect the plastics in a different container to the regular refuse. But the garbage men come and they will put all into the same thing. So why am I separating it out?
Even your governance is also one of the issues there.
Shivraj, the process of self-control, I believe, has already started. If you look back 2 years or 3 years before, we were very happy to get the plastic bags when it was given free of charge. And even somebody used to ask for an extra plastic bag. I don't know why, but we used to ask. But gradually, the departmental stores are realizing that, okay, it is a serious threat. Even the standard shops in our neighborhood, they are also not keeping it. So the process has already started and people are realizing that it is a serious problem, even though probably they do not understand the dimensions of it. So definitely things will improve, but at the same time, there are a couple of issues we need to address now. One issue, as I mentioned, that okay, adequate amount of consolidated research must take place. Because those research findings are ultimate ways of convincing people that this is really going to be bad. Second thing is that we are going for a shortcut, that biodegradable plastic, without understanding that during the biodegradation, what comes out. It may be microplastics, it may be some chemicals which have got a very serious carcinogenic effect, but those serious studies have not been done. So our basis should be formulating a very strong research program, and I would request you to, the way we have discussed, to start the process. And at the same time, so far the general public is concerned, it is necessary to tell them through very simple documents that what are the best practices for handling the problem. So maybe some of your group in the environmental department can also work on this in collaboration with other agencies. Yes,
Thank you, sir.
Sivat.
Thank you, sir.
Okay, now John, more or less we are coming to an end. If there are any.
One comment first, and that is, we also have plastics in Norway. So I just want to let you know, and fortunately in my community, and that's only my community, we have a very advanced collection system. It's not true for all over Norway, but in my community we have one. And that is, we are mandated to put the garbage in plastic bags, like the one you get in the shop. And they also give out green plastic bags, which we have mandated to put the food waste in. So when everything then we collect everything into one bin. So when it comes to the garbage plant, there are lasers that first sort out the green bags and take them to a biogas plant. Then the rest of the plastics, it's sorted by their reflection, so they can determine which type of plastic it is. So then they do the fractioning of the plastic automatically. And I think now, having a very advanced technological waste management system, that's the way to go. Because I think it's possible to ban plastic, and we don't want to do it either. So we want to have plastics as bags, even though people tend to kind of dispose of them. The main thing is then to get them back into the system again. And for bottles, we do have a deposit. So every time we buy a bottle, a plastic bottle, we have to pay an extra amount. And then when we return it, we get money back. And this means, you will never see a single plastic bottle in Norway thrown around, because people collect them. Even if you don't use them yourself, there will be someone else that picks them up, because they get money, like 20 cents for each bottle. And then it's not only beverages, but they also started to use it on all kinds of plastic bottles, with liquid, with soaps and everything. So I think that's one good way to be able to motivate people to return their used bottles. Okay, are there more questions?
Jon, Neville, yeah. So those two remarks now are very interesting to me. My experience in South Africa is in the neighborhood that I live in. Indeed, there is refuse collection, but not sorted in any way. It all goes to landfill. So I separate voluntarily. I separate out my plastics, and I put them in my vehicle, and I drive them once a month to a recycling depot. But that's voluntary. Down the road from me, as I said this morning, 2 or 3 kilometers down the road, there's no refuse collection at all for any kind of refuse. And that refuse just accumulates in the poorest of areas. And it just gets worse and worse and worse. And people live amongst the refuse. And I agree with you both. One solution is to look for alternatives, economically viable alternatives to plastic. But that's the end of the problem. The problem needs to be dealt with at its source. And I do believe incentivizing people to use plastic or recycle plastic is really an important solution. And Jan, you've just explained one way to do it. In South Africa, the poorest people get an allocation of electricity for free from the government. They get an allocation of water where water is supplied for free. I think similarly, we should incentivize people to conserve plastic. And one way is indeed, right now, if I collected 20 kilograms of plastic and I took them to a company that recycles, I can get some money back for that. But that's because they make money out of recycling the plastic. If there was a charge on the use of plastic and a value put on the waste, if you put value on the waste, then the waste will become a currency. And that is one way to incentivize people. So I totally agree with that approach. The problem is a social and economic one as well as a technical one. Thank you.
Thank you. So if there are no more questions, then we should get to the close of the panel discussion. So could we have Suri and Shivraj? There is only one item remaining in the total program, that is the drawing the resolution. So we'll start after this. But before we do that, it is, see, this program has been possible with the planning and entire execution by all of you, especially Shivraju and Suryanarayanan. So on behalf of the NAM Center, it is my pleasant duty to present them a small memento. So I request both of you, they can come, and I'll request John to hand over the memento to them. Thank you. Break down, sorry, only from downstairs, the resolution from the project. Thank you so much.
So thank you, Dr. John, for chairing that wonderful panel discussion. I also thank all the panel members for their active participation. Thank you. So now, we are going to adopt the resolution. Now, actually, we have circulated the resolutions, and there are feedbacks and suggestions. So now we are going to project the resolution, and
tea break.
Okay, okay.
After that, start the resolution session.
Okay.
Now we can tell them that we will start the resolution session at 4:15.
Okay.
It will take half an hour. 40 minutes.
Okay, there is a small change in the evening. So now we can we can go for tea break. Okay, followed by adaptation of resolution. So we are going to reassemble here at 4:15. Thank you.
Thank you.
I request the participants to please reassemble and please settle down. I request Dr. Amitava Bandyopadhyay, Sir, Director General, NAM SND Center, India, to please initiate the resolution activity.
Thank you, ma'am. We are coming to the last leg of the seminar, and this is a session called resolution session. And many of you may ask that exactly what is the resolution. Now, these are a set of technical recommendations which are discussed and taken by this scientific group. And Dr. Suryanarayan and Dr. Shivraju and their team, they have prepared a draft resolution, and yesterday a copy was circulated to many of you. And based on your subsequent comment, the document has been revised. And what you see in front during the next few minutes is the total document projected on the screen. During the session, we also again give an opportunity for the public to express their views in case still there are some minor corrections to be done. Now, one thing, let me assure you that this resolution document is only from a group of scientists and technologists. This is not a view of the government of the participating countries. So essentially, this is a non-binding document. But then again, you can ask that if it is a non-binding document, what is the use? Many times in NAM Center, along with our partners, we venture into an area which is practically unknown to many of our member countries. So if they have to prepare a roadmap for that particular subject, they need some amount of guidance. So essentially, the resolution becomes a guiding document for them. So with these few words, again, I am repeating, this is a non-binding document, and I'll request Professor Sumana to start the process. Thank you.
Thank you, sir. On behalf of the organizing team, I shall be just reading out the resolution regarding the international workshop on combating plastic pollution in terrestrial environment. Shall I start, sir? We, the participants of the international workshop on combating plastic pollution in terrestrial environment held at Mysuru, India during 14th and 15th March 2023.
Plastic pollution. Okay. The international workshop should be deleted.
Okay.
International workshop on to be deleted.
Okay, sir.
So it is Mysuru resolution on combating plastic pollution in terrestrial environment. You can make the change. Yeah, you have done. Okay. Now it should be reflected there.
Mysuru resolution on combating plastic pollution in terrestrial environment. Sir, in the title, Mysuru resolution on Combating Plastic Pollution in Terrestrial Environment.
That will be that will be. Yeah, let's see what is probably there. No problem. Okay. Okay. But no, I have. Okay, Mysore. Then should you make that OL capital? Okay. Now, in the first line, will the participants of the, here the full thing will come, international, so that Mysore will not be there. Will the participants of the international workshop on the resolution, yeah, that's fine. Will the participants of the international workshop on combating plastic pollution in terrestrial environment held at Mysuru, India during 14th and 15th March 23. Wonderful. Yeah. Ma'am, you can continue, please.
Thank you, sir. Representing the governments, academic and research institutions, and SNT agencies, as well as other relevant stakeholders from Egypt, India, Indonesia, Iraq, Malaysia, Mauritius, Myanmar, Nepal, Zambia, South Africa, Sri Lanka, and Norway.
The Iraq will go out.
Okay. Iraq.
Because the delegates got here. Iraq and Zambia. This is okay. This is fine.
Okay.
This is edited. It's already.
Okay, sir. Yeah.
Yeah.
Shall I proceed, sir?
Yeah, please.
Recognizing that plastic pollution has become one of the most pressing environmental issues in the world that affects terrestrial, aquatic, and marine ecosystems, and that accumulation of plastic waste in the terrestrial environment is a significant threat to wildlife, human health, and ecological balance. Realizing that concerned efforts to combat plastic pollution in the terrestrial environment is crucial for the health and sustainability of the planet. Recalling the aims of the workshop to bring together experts from NAM and other developing countries in pollution prevention efforts and related ecosystem and services and socio-economic sectors, as well as members of planning and policy groups working towards combating terrestrial plastic pollution. Reaffirming the commitment to combat plastic pollution in the terrestrial environment in NAM countries to ensure that the efforts to address this crucial issue should be made with clear and measurable goals, increased funding, participation of stakeholders, innovation, and monitoring of the progress. Unanimously resolve and recommend the following. Policies and strategies should be adopted to reduce single-use plastic products such as straws, plastic bags, and water bottles, and encourage the consumers to switch to reusable alternatives such as reusable water bottles, cloth bags, and metal straws.
Can you just go back?
Unanimously resolve and recommend the following. Before this one, sir.
Which part of page one?
Reaffirming. Go back. Reaffirming. Okay. Recalling the aims. Recalling the aims of the workshop to bring together experts from NAM and other developing countries in pollution prevention efforts and related ecosystem and services and socio-economic sectors, as well as members of planning and policy groups working towards combating terrestrial plastic pollution. Reaffirming the commitment to combat plastic pollution in the terrestrial environment in NAM and other developing countries to ensure that the efforts to address this crucial issue should be made with clear and measurable goals, increased funding, participation of stakeholders, innovation, and monitoring of progress. Unanimously resolve and recommend the following. Policies and strategies should be adopted to reduce single-use plastic products such as straws, plastic bags, and water bottles, and encourage the consumers to switch to reusable alternatives such as reusable water bottles, cloth bags, and metal straws. Governments, business, academia, R&D, and individuals should contribute to reducing plastic pollution by implementing proper waste management practices, including recycling, waste segregation, and disposal of plastic waste in landfills. Governments, non-profit organizations, scholars, and schools should organize campaigns to educate the public about the dangers of plastic pollution and the importance of reducing plastic use. Businesses should be encouraged to use sustainable packaging and opt for eco-friendly materials such as biodegradable or compostable plastics, paper, and cardboard. Governments should impose ban on the production and use of single-use plastic items such as plastic bags, straws, and utensils. Governments, businesses, and non-profit organizations should invest in research and development to find alternative materials and products that are biodegradable or compostable. Regional level development and implementation of a policy framework should be facilitated. Governments and all other stakeholders should encourage and support the United Nations commitment to reduce the amount of plastic that leaks into the environment through a number of projects that focus on microplastics, marine litter, and single-use plastic pollution that can be a catalyst for change across the developing world in regard to Agenda for Sustainable Development 2030. Combating plastic pollution in a terrestrial environment requires a concerted effort from governments, businesses, NGOs, and individuals. Implementing a combination of the above solutions would help reduce plastic pollution and ensure a cleaner environment for future generations. Increase pressure on manufacturers to oblige to ERP by responsibly recollect and recycling the produced plastic and e-waste.
Can you use the full talk?
Pardon, sir. Repeat, sir.
Just explain the term ERP. I think we have not used the term ERP earlier.
Yeah. Okay, okay, sir.
So, Shivraj, what is the full? Yeah. Okay, extended producer responsibility. Bracket ERP. D.
increase pressure on manufacturers to oblige to extended producer responsibility by responsibly recollect and recycling the produced plastic and e-waste.
EPR, okay.
That this resolution may be submitted to the heads of state and governments of NAM and other developing countries and other participating institutions for appropriate actions. Thus unanimously resolved and adopted on this day, the 15th March 2023 at Mysuru, India.
Yeah, the T should be small. Yeah.
Thank you.
Thank you. Thank you so much.
We are waiting for Vice Chancellor and our Register to start with the validatory function. I request everyone, all the delegates, to please be seated and have patience. They'll be coming within 5 to 10 minutes.
This campus is really pretty because its greenery it has been maintaining. The faculty is amazing here as they are very understanding. They provide great encouragement in whatever we do.
I'm proud to be a part of this prestigious university.
I've been proud to be a part of this academy which serves the society through its services and education. I would definitely recommend this subject and Basis Academy to my juniors and my friends. International Student Hostel. And I would like to encourage any international student to join this college. This is what I expected from my bachelor's life.
Sciences, where all life sciences related research levels are provided.
This campus is really pretty because of its greenery it has been maintaining. The faculty was amazing here as they are very understanding. They provide great encouragement in whatever we do. I'm proud to be a part of this prestigious university.
I feel proud to be a part of this academy which serves the society through its services and education.
I would definitely recommend this subject and Basis Academy to my juniors and my friends. International Student Hostel.
And I would like to encourage any international student to join this college.
This is what I expected from my bachelor's life.
Life Sciences, where all life sciences related programs at the undergraduate, postgraduate, and research levels are provided.
ಯಾವುದೋ ಯುದ್ಧಕ್ಕೆ ಬಂದಂತಿದೆ. ಯುದ್ಧ ಅನಿವಾರ್ಯವೇ?
Good evening, all. So now we came to the end of the session. With the divine blessings of His Holiness Sri Shivaratri Deshi Kendra Mahaswamiji. So we came to the today's end of the session. Now I request Dr. Pallavi N, Assistant Professor, Department of Environmental Science, to welcome the dignitaries.
Offering my respectful pranams to the Suttur Shri Kshetra, I am delighted to welcome you all for this valedictory function of this two-day international workshop on combating plastic pollution in terrestrial environment. I am very delighted to welcome our Vice Chancellor, Dr. Surendra Singh, for this valedictory function. Welcome you, sir. I am pleased to welcome Dr. Amitav Bandopadhyaya, Director General NAM S&T Center, who sat with us for all these 2 days in workshop, giving his valuable input and interaction with us. I welcome you, sir. I also welcome John Samseth, President Scope Netherlands, who was also with us, giving valuable inputs and interacting with us. Welcome you, sir. I also welcome the backbone of this workshop, Dr. Vishal Kumar Gupta, Dean Academics, JSS AHR, for this function. I welcome Dr. Surya Narayan S, who has really worked hard with us, collaborating, and he was very much helpful for the successful of this program. Welcome you, sir. I welcome Dr. Shivraju HP, who had worked day and night for the success of this two-day international workshop. I also welcome all the principals and vice principals of our constituent colleges, heads of the department, and all the administrative staff of constituent and department colleges for this valedictory function. Last but not the least, I would like to welcome all the participants, delegates who have been with us for these 2 days international workshop, interacting, giving all your valuable inputs. And without you, this function wouldn't have been successful. I welcome you once again. Thank you, Ananda.
Thank you, Dr. Pallavi. Now I request Dr. Vishal Kumar Gupta, Dean, Dean to give opening remarks.
Hearty good evening to all of you. I submit my pranams to the lotus feet of His Holiness Swamiji, and I welcome our Vice Chancellor, Dr. Surendra Singh Sir, Professor Yon from SCOPE, Dr. Amitabh Bandyopadhyay from NAM SNT Center, who has been instrumental in guiding us and conceptualizing this workshop, sir. Also welcome to Dr. Sur Narayan, as well as Dr. Shivraju and his entire team, participants, students, and dear friends. Well, any good thing has to come to an end, and this is a valedictory function, and I believe that you would not have realized how the 2 days have gone through, how a lot of deliberations have happened, and we have got amongst us lot of eminent speakers who spoke on not just the policies, procedures, the ideas, innovations, but also have shared their practical experiences on how to combat this plastic pollution, which is a big problem in the present world. JSSAHR has been at the forefront in collaborating and especially envisaging its vision and mission towards the healthcare needs of the national as well as international forums. So with this direction only, this workshop was also conceptualized with a blend of sciences, and in that focusing especially on the health. I'm very glad that SCOPE and NAM SNT Center have provided us an opportunity to collaborate and organize this workshop. I believe a lot of friends whom have made over these 2 periods of 2 days of time and from 12 different nationalities will have a wonderful experience in this. I must congratulate all the entire team for the successful organization of this workshop, and the resolution which was formulated just prior to this valedictory function, I believe will go in a long way in addressing and providing guidance, especially to the combating plastic pollution, because it is going to be circulated to various regulatory bodies throughout the world. With this, I conclude my speech and thank you one and all.
Thank you, sir. Now I request Dr. John Sanchez, President of SCOPE, to address the gathering.
Thank you. Dignitaries, professors, and students. It has been a very nice experience for me to be here, because this workshop is going to be significant. It's going to contribute to the understanding of the terrestrial plastics situation. The focus has been on terrestrial plastics, but it has also been without overlooking the ocean plastics. So for SCOPE, this has been very successful, and we have brought many new faces into the SCOPE network community. And SCOPE is actively seeking members that can contribute to our scientific agenda. And I'm quite sure that many of you in this audience sooner or later will be invited to join our panels. And I'm very encouraged to see so many young scientists here, because they will be the future. And I can see by the level that has been presented here that it is not a bad future we are going to face within science. So I think the use of advanced techniques and working on advanced problems. So I'm very glad that we have been able to have a successful workshop with the JSS and the NAM Center. And hopefully this will be one of the many workshops we'll have together and a number of publications that we put out. And then particularly thanks to Suri and the Sedrash for the excellent hosting and organization of this workshop. Thank you.
Thank you, sir. Now I request our Honorable Vice Chancellor Surinder Singh, JSSAHR, to address the gathering.
A very good evening to all of you, and I think it's a proud privilege for me to be part of this latest session of this international session on workshop on combating plastic pollution and terrestrial environment, which has been jointly organized by JSSHR, NAM SNT Center, and SCOPE. At the outset, I would like to submit my respectful pranams to His Holiness Swamiji, because this program has been a success and we have been able to do this program with his blessings and all the support which we have been receiving from all our authorities. But dear esteemed all faculty members, honorable guests, honorable delegates, and dear students, this momentous occasion and the momentous event of which we are having the valedictory function today is of utmost importance to each one of us, because plastic pollution, it touches the life of each and every individual. And I think I feel very proud of all our faculty, all of our students who have worked very tirelessly to ensure that this workshop becomes a success. And I thank all the delegates who have come from the different parts of the country and from the different parts of the world also to do it. This workshop is basically a testimony of our university's commitment to promoting environmental sustainability and global outreach. As I mentioned yesterday, that the most important thing is we can have these seminars, we can have workshops, we can have any number of literature, booklets brought out, which have got their own importance. But most important thing is action, action, action. We need to act, then only the changes will come across over it. And I'm very proud that our JSSHR has taken a significant strides in internationalization, which has made this workshop a possibility and a reality. Our university believes that a global approach is essential to address the environmental challenges that we face today. And the international exposure that such events offer provides a platform for learning and sharing of best practices to achieve the SDG goals of the United Nations. The theme of the workshop, combating plastic pollution in terrestrial environment, is of the utmost importance in the present context. This workshop has provided us with an opportunity to discuss and deliberate on not only the challenges, but also the way forward. And we also, from this workshop, will be taking home message that how we should be combating the plastic pollution. The most crucial takeaway is the need for collaboration and a concerted effort to tackle this issue. And it is not just the responsibility of the government, but it is also the responsibility of each and every individual to take action. And this is the reason when I was basically here on this platform yesterday, I said that the first and the foremost thing which we will do from yesterday onwards itself, not today, because I think in the program it is written that we will take a pledge. We already had taken a pledge yesterday, and when we went back to office, our registrar has already initiated the action. And this is what our board of management members also tell me again and again, that what we have seen in JSSHR is people do what they say. And the biggest testimony in this regard is yesterday itself when we went over there, and I was surprised and I was told by my officials in the building in which I said, the registrar had come and given instructions that henceforth we will see to that all plastic is done away in our building. And the exercise started yesterday itself. The thing which really touched me was there was not only the registrar, even my personal staff came to me, all the people who work in that division with me, they also came, sir, we will work it out that how we can basically phase out plastic. And the first and the foremost thing which we want to phase out is plastic bottles and to have these glass bottles. And already we are working on where to put the RO system, where to get the bottles, how to put the systems in place. And I think all the concerned colleges, departments, and the principals will also be taking in action, because individually I asked for the pledge today, because what happened? Individually I interacted with principals pharmacy college, with the principal of the medical college, dental college, and they all committed. They said we will do it also. So I think that speaks more than a pledge. Taking a pledge is one thing, but really putting the things into practice is another thing. And I think next time when Dr. Amitabh Bandyopadhyay will be here, Dr. John Sampson will be here, we will show them around and very proudly that you see our JSSHRS campus is plastic free to the extent possible. There might be some things which we cannot do with, but 99% or more than 90% will see to it that our campus is plastic free. Why I say this thing is all the delegates, all the students who are over here, as I said, it's not a government responsibility, it's a responsibility of each one of us as a responsible citizen. And a citizen responsible even for our future generations, that we should take action in this regard and do it also. And from our side, from JSSHR side, I can assure NAM-SNT and Scope, Dr. Jan Samsat and also Dr. Amitabh Bandyopadhyay, that we will come up to their expectations. And this workshop, this is the one of the, I think, the initial steps in that direction. And I want to reiterate the importance of this workshop by saying that by promoting this environmental sustainability by our joining of hands, what we are trying to do is give a strong message to other participating countries in the region also, so that they also can take the steps in this direction, and eventually we can see to it that we can minimize or to the extent possible the use of plastic in our day-to-day life. So with these words, I thank Dr. Bandyopadhyay, Dr. John Sampson, Dr. Suryanarayan, Dr. Shivraju HP, Dr. Vishal Kumar Gupta, our Dean of Academics of our FLS, Dr. Ravisha, and all the students of the Environmental Sciences Department who have worked tirelessly to ensure that this workshop is a success. And to the students, I would like to make appeal that you please take up some initiatives which have been discussed in this at your personal level, and also try to see that how you can implement at your end over here. Because you are tomorrow's future environmental leaders, and you are the one who will be taking this lead and taking this our efforts further in this direction. So I wish you all the best, good luck, and let's make our JSSHR campus, our Mysore city, our country, and entire globe plastic free in the times to come. Thank you.
Thank you, sir. Now I request our Honorable Vice Chancellor to felicitate Dr. Suryanarayan, sir. Thank you, sir. Now I would like to request Dr. Amitabh Bandyopadhyay, Director, NAM-SNT Center, New Delhi, to give closing remarks.
For such a diversified workshop to be closing remarks is a very difficult job. So I want to try that. Pronouns to His Holiness Swamiji. I take this opportunity and the JSS Senior Management, Dr. Suresh, the Chancellor, Dr. Sundar Singh, Vice Chancellor, Dr. Latha, Registrar, Dr. Ravisha, and all the Action Assistant, and all other senior officials for their outstanding support in regard to this workshop. Thank you so much. I take this opportunity to thank John and Neville, who should have been here, but because of certain visa delays, he could not come. Thank you, John. Thank you, Neville, for your great support in organizing. You are planning it for many months now, and we have always received outstanding support from the Scope senior officials. Now, plastic pollution, as many of you have understood after we have seen all the presentations and seen the poster presentations,
that the subject is extremely complex. On one side, we have the problems about the logistic problems, the best practices issues. So on one side, that should be tackled, but being academicians, being researchers, it is our duty to set up a good research program. And that's during the concluding session we have suggested. And I again propose in presence of Dr. Singh that in fact it is a reiteration of what he said yesterday, that there should be a proper research center dealing with this problem. And sir, with your support, maybe you will get the support from the Karnataka Pollution Control Board, because I'm quite confident that when Dr. Thimaya was here, he was also impressed by the things that he has seen here. So may I propose that we'll initiate the process of starting a joint JSS-AHER scope and NAM-SNT Center of Excellence on doing research on plastic pollution. And I am hopeful that John will agree to that. So if we start today, it will maybe take 5 years for the center to come to a shape. What I have also suggested to Dr. Shivraju that initially that center can be a virtual center for sharing information, because that doesn't require much resources. And as we grow, as we become more matured in our understanding of the issues, we can start the physical center. Because there is a growth scope for both contribution and collaboration. We thank all our delegates from different NAM countries and other delegates for being with us today and for these last 2 days and for very, very active participation. I thank my colleagues from the NAM-SNT Center. They have done an outstanding job. Thank you. Then again, it is my I would like to thank once again Dr. Shivraju, Dr. Suryanarayan and Vishal. The way they have planned it, they have executed the program and ensured that it comes to a very, very successful conclusion. Is outstanding. Thank you. And finally, my sincere thanks to the team JSR, team JSS, because without your active participation, support, it would not have been possible. So thank you so much once again. Thank you.
Thank you, sir. Now I request among the delegates to give feedbacks about the international workshop.
This workshop is well organized and managed. I really appreciate the program. The workshop is well managed and organized in the beautiful city of Mysore, India. And really appreciate. I will take the message to my country.
Any other persons who wish to share their experience about the workshops they can?
Yes, as you can see, we have learned a lot from the international workshop. Thanks to GSS NAM-SNT scope. As my colleague said, we will try to bring what we have learned to our countries and try to implement. As VC said, we have to have action, action, and action. Okay, thank you.
So actually, this workshop was 2 days we enjoyed and we learned a lot. And which research activities as an R&D institutions we have to continue for the combating the plastic pollutions. We got several ideas and in our country, so we have to follow these research ideas to further combating these plastic pollutions to home the future generations. Thank you very much for.
I think this workshop it's been an excellent workshop. We were able to get a holistic view of the problem and all the possible solutions. That is the most important thing for us because each one of us is working on one aspect. So it is very important that we are able to get the view of all the other aspects of it. And that was one thing that I realized we got here. So thank you very much for that.
Thanks to all the delegates who shared your valuable experiences. Now, I request Dr. Shivraji HP, Associate Professor, Department of Environmental Science, to propose a vote of thanks.
Good evening, all. Seeking the blessing of His Holiness Swamiji, it is my privilege to propose the vote of thanks on this occasion. First and foremost, I would like to thank Dr. C.G. Betsurmat, Executive Secretary at JSS Mahavidyapita, and Dr. B. Suresh, Pro-Chancellor, JSS Academy of Higher Education and Research, for their continuous support and encouragement. I express my heartfelt thanks to our Vice Chancellor, Dr. Surinder Singh, who is with us throughout the program and guided for this successful of this event. Thank you, sir. I would like to thank Dr. Amitava Bandopadhyaya, Director General, NAM SNT Center, for his continuous support, guidance, and encouragement. Thank you very much, sir. I would like to express my sincere thanks to Professor Jan Sanset, President of Scope, support and thank you very much for support and active participation through online. We are very grateful to our Dean, Academic Dr. Vishal Kumar Gupta, and Deputy Dean Academic Dr. Prashant S. for their continuous encouragement and support for success of this event. Thank you, sir. I sincerely thank Dr. Suryanarayan S., who is with us all the way and guided us, suggested that, and he was the part of event. He spent most of his time to organize this wonderful workshop. I use this occasion to thank Suryanarayan S. Thank you, sir. I thank all the international and national delegates for their interest and sharing their thoughts during this workshop, and all of them have actively participated, and they shared their knowledge, and especially to solve plastic waste management. Thank you all. I thank our registrar, Dr. B. Manjunatha, for his motivation and encouragement for this event. I also thank all the university authorities, staff for their direct and indirect support. I would like to extend my sincere thanks to Dr. Ravisha K., Dean, School of Life Sciences, and faculty members, my dear colleagues, students, for their continuous support and active participation. I would like to extend my sincere thanks to Principal, Administrative Officer, supporting staff of JSS Medical College for providing this wonderful auditorium and other logistics. I also thank NAM SNT Center team for their support. I thank all media person for their presence and wide coverage. I thank my dear student, fellow research scholars for their cooperation and making this function a great success. Thank you, one and all. I also request all the participants join for group photograph. Thank you.